Okay, everybody. We're going to start our first talk session. I hope everyone had a good breakfast. I'm putting some more announcements. I realize there's probably some confusion. Like the grad lounge Surfside's a little crowded, so feel free to eat in that courtyard area in the morning. I posted a picture. I'll post that where more bathrooms are. So they're all in the back there. There's also 2 bathrooms that are close to Surfside. But anyway, let's get started. So our first speaker is going to be Gage Siebert from ASU.
Hello, everyone. Yeah, so for those of you who haven't met me yet, my name's Gage, and I'm going to be talking about assembly theory, and in particular strings. And hopefully by the end of this, I will have convinced you that it is very useful for astrobiology in particular. So in assembly theory, the primary quantity we're concerned with is called assembly index, and it's a complexity measure that corresponds to the minimum number of joining operations that it takes to build an object. So if my object is like X, and I need at least 3 of these steps where I combine 2 things to make something new to get to X from some set of fundamental building blocks, then we say the assembly index of X is 3. And so assembly index is kind of system dependent in much the same way that entropy is, where for entropy I get to choose what my micro and macro states are, and this influences exactly what it means. And for assembly index, these things that we get to choose are what our fundamental building blocks are and what the different joining operations that are allowable are. And so we kind of philosophically what this thing means is that if I find some really complex object, let's call it A, and it has an assembly index of n, and I find lots of copies of this object, I can be sure that somewhere in the environment there's at least some n construction processes that exist there. So that memory is stored somewhere in the environment that's creating these objects. And that just simply has to be true because for objects even as small as like modest sized proteins, there's not enough like matter on Earth to make all of them in any kind of meaningful abundance. And so this approach kind of lives between 2 different worlds. So one is like, I might want to know the explicit physics or chemistry that's going on and know exactly how these things are actually built every time and understand those pathways. But in assembly theory, we just take what's the minimal like hypothetical thing that we can't rule out as impossible. And this is a massive simplification, so we don't have to understand all the microphysical properties, but we can still put lower bounds on how much it can take to make these objects. And on the other end of the spectrum, it kind of varies from algorithmic information theory, where I would instead say, something is so simple based off of how small of a program I can write that creates it in a computer language. But in assembly theory, we're requiring at every step that everything that's happening is physically possible. And so strings are biologically relevant, as we are all probably quite familiar with. It can be encoded in like 2 different alphabets, so I can write it in like nucleotides or in amino acids. And like many other fields of physics, studying strings is like studying symmetries. And so this is kind of a diagram here of this top string of where different symmetric substrings are within it. And when you look at this, these correspond to meaningful properties in the ways you can build it in the shortest paths. So one feature I want to point out is that, so in this bottom left bit here, I have this 1-2 segment that I'm doubling with itself into 1-2, 1-2. And we call this kind of innovation-like, in that it's like I have the blueprints for it and I don't need to rebuild it from scratch every time. So this has a lot of really nice mathematical properties, and it's a really rich subject. So like this thing on the bottom looks like ice cream with sprinkles in it, but it's some kind of weird graph which actually corresponds to like how you reconcile all these symmetries together to calculate like what the shortest build path is, and it has a really nice relation to some like really classic problems in computation theory that tell us about how complicated it is to calculate these things. But much more salient to astrobiology is thinking about what we can call prime strings, or I would argue also materially efficient technosignatures. So the example I want to kind of describe this as is it's just a series of numbers, but what would it mean in maybe a material is if I restrict myself to working with polymer chains, And my alphabet size is basically how many different monomers I have access to. If I want to find how do I efficiently create something that demonstrates I know how to compute a lot of different reactions, then I need something that has as few symmetries in it as possible. And there's actually really explicit ways to construct these things such that they have absolutely no, I can construct, there's many different ways to build these strings, but none of them reuse the exact same reaction twice by virtue of just what they are. And that's a property intrinsic to these objects. And this also kind of highlights the difference between this and algorithmic information theory, because like, if I asked anybody to like make the next one, you'd probably all get it right on the first try. There's a lot of regularity in what these objects look like, and so algorithmically they're not that complicated, but in terms of the physical reactions you need to make them, they really are. So something where I see this going in the future, and that what I'm really excited to do with this is have a dynamics of how life can explore the chemical space of what's possible. And so the picture I want to illustrate, these are not real data, this is just to illustrate a point, is I want to think about some space of objects, and I think a good example is all the proteins that a species creates, say. So we take all of these proteins, and I want to assign distances between them all, and qualitatively, I mean, there's an example of how to do that on the top, but qualitatively what we should think of is the distance between x and y is the number of joining operations that I need to change to go from one to the other. And so things that are intuitively similar will be closer, just, yeah, as you might expect. And so this is really useful because when we think about what we have as a generative model for like a null hypothesis, like what should life be doing, we know that mutations are things that really only change assembly index by one. Either I'm inserting some large segment that already existed, and so I didn't have to build it from scratch, or I'm making some really small change. And in any case, this is a change in assembly index of just one. And so we have this kind of thick surface of the adjacent possible, like things you can mutate into. And because it's reasonable to imagine that this is happening kind of randomly, we can have some kind of null model for what we expect life to look like. Like, what's the shape of this object as it grows in time? And so I like to think of it as kind of spherical, but actually it's probably a little bit noisy just because it is so random. But in reality, I don't think we'll really observe this because some mutations will be selected out and we'll never observe them. And so the way I want to illustrate this is this: you have this kind of noisy sphere on the left, and you have a much noisier version of it on the right. And if I observe this for two different things, the interpretation of this is we're seeing something about like the fitness landscape. Like this is kind of the shadow that makes it through, right? So like there's lots of voids here of things that are either not functional or even hurtful to the system. And by studying like the geometry of these kinds of objects and looking for the ways they scale, we can learn things about the ergodicity of life, meaning if I were to restart us from like early Earth times, how similar would I expect the biochemistry to be today that we observe? Just by virtue of how many ways does it seem like there are to actually move in this space? And by extension, we can make inferences about just life in general and how constricted it must be and how diverse will potential alien biochemistries be. So with that, I'll leave you with a fractal that reminds me of the building process, and I'll take any questions.
So how can this be employed to sort of create a criterion for what constitutes abiogenesis based off the assembly complexity?
So there is an excellent Nature Comes paper, I want to say from 2020. It's Marshall's the first author, where they talk about, so you can do this for molecules, and they find a really big disparity between what kinds of things abiotic sources can produce versus biotic. And this is something that you can measure quite accurately with like mass spec and some other methods too. And so just for a chemical biosignature way, there's a concrete thing that already exists.
Thank you.
Yeah.
Hi, great talk. And I was also just curious, when you're evaluating the assembly theory, is this based off of entirely the sequence, or do you also account for structure?
I don't
Like
understand.
protein folding or something?
Yes, so this is kind of embedded in like our choice of the definition of the joining operation, right? So like the simplest choice doesn't count the protein folding, but you can imagine that I define the joining operation such that I allow myself to consider those like higher order constructions. And so yeah, it's a choice of your specific instance, yeah.
Hi, yeah, this is very interesting. I'm wondering, how do you consider the starting amino acids you have? Because there's a lot of theories on like what amino acids were like first, or like which evolved later, and like, are you only using the canonical ones that we have in like the current genetic code, or like there any like theoretical amino acid structures that you're exploring?
I'm not. There's someone at your table who does. his name's Thomas. But to relate it to this, I think this is kind of embedded in your choice of like what the fundamental building blocks are. It's like what's your T0? Yeah.
Any other questions for Gage? Okay, let's give him a round of applause. Okay, and our next speaker is going to be Alex Plum here from UCSD.
Hi there, I'm Alex Plum. My talk is on autocatalytic chemical ecosystems in spatial settings. So I wanted to start with this quote from the French naturalist Georges Cuvier. I don't think it's a good definition of life, but I think it captures one feature of life that's very important, specifically this feature of autocatalysis, which I think is the central motif for self-propagation that we see kind of all across life and that may have been relevant in life's earliest stages. So we say that a process is autocatalytic if the products of that process catalyze the process itself. Here I'm going to be talking about autocatalytic cycles, where you have a sequence of reactions that form a cycle, such that with every turn of the cycle, you get a stoichiometric increase in the number of some set of chemicals that we'll call member chemicals. You can take all of the chemicals involved in an autocatalytic cycle and partition them into food chemicals, into member chemicals, and into waste chemicals based on which sides of the reactions they show up in. And so here's a simple example where you just have two reversible reactions. You have two member chemicals, M1 and M2. You have one food that's being used for both reactions and one waste chemical. And in principle, these can be irreversible. Here I've drawn them as reversible. And because they're reversible, you can drive them in either direction. If you have an abundance of food, they can be driven in the productive direction that provides the stoichiometric increase in the member chemicals, allowing them to self-propagate. But if instead you have an accumulation of waste, it can kind of wind down in the other direction, and you can get a stoichiometric decrease in the number of member chemicals as well. So we've analogized these autocatalytic cycles to biological species, in part because they're consuming food, they're producing waste, and self-propagating themselves, and we've demonstrated that they can exhibit logistic growth. If you put them in a closed reactor, some amount of food available, some seed member species, they can start growing and they can reach equilibrium concentrations. But this isn't of great interest in an origins of life context because life is an out of equilibrium process. So in practice, we typically simulate these cycles in chemostats, wherein you have an inflow of food from a source that drives them in the productive or autocatalytic direction, and where everything is diluted out of the system. And this has two implications. One, it helps to offload the waste, to kind of keep it from being driven in the other direction. And it also provides a selective pressure, so that now if the cycle doesn't replicate or propagate its member species quickly enough, they'll just be diluted out of the reactor. So it's not trivial that these cycles are going to persist anymore. Persistence becomes a problem, something that they have to kind of achieve. So when you simulate these, you can get time series that looks like this plot over here, where you have a depletion of food through time, you have logistic growth in the concentrations of member chemicals here in blue, and you also have a buildup of waste. You can choose to simulate them using ordinary differential equations if you're in a well-mixed reactor, and that assumes that you have large enough quantities that you can treat continuous concentrations of chemicals. The other approach is to simulate things stochastically, and in kind of the large n limit, this looks very similar to the continuous case, but as you go to smaller and smaller numbers of chemicals, things look a lot noisier, and you can start to have more contingency in the dynamics, where you can have the stochastic loss of a single chemical or the stochastic dispersal of a single chemical in a new location. changing the dynamic somewhere else. And so in everything I show here, I'll be taking this stochastic approach. So that's all the dynamics for a single cycle. We're ultimately interested in combining different cycles and seeing how they interact ecologically. So here I show the member chemical concentrations over time, or counts over time, for the different autocatalytic cycles shown on the left here. They can exhibit competitive exclusion, competitive coexistence when they share a common food source. They can also exhibit mutualisms, wherein the food or the waste of one chemical serves as food for another autocatalytic cycle. They can also exhibit predation, wherein the member chemicals of one cycle serve as the food for another cycle. And the dynamics resemble those that you get in the Lotka-Volterra equations in ecology, where you can get both stable and damped oscillations, predator or prey dominance, or coexistence of the predator and prey. And you can also get priority effects, wherein two cycles might mutually inhibit one another, so that it matters if one cycle gets seated in a location first versus the other, since they'll suppress the growth of the other cycle. So why is this relevant to the origin of life? We think that these autocatalytic cycles provide one of the simplest ways, in the absence of needing compartments or polymer genetics, to get this sort of self-propagation that you see in metabolism. And we think that they can provide an avenue for the accumulation of complexity to ultimately serve as precursors or scaffolds for later stages in the origin of life. So as an avenue for kind of increasing the chemical diversity that you might need to achieve later stages. And we think that you can do that by taking individual cycles, composing them through various ecological interactions, and then using the sorts of avenues that we see in ecology for the accumulation of complexity. So how do you accumulate complexity in ecology? One way is through ecological succession, wherein you have some species that lay the groundwork for others to later succeed them. You can imagine this in a chemical context where the waste or member chemicals enable the activation of new cycles that couldn't have been activated until those first cycles were activated. You also have ideas in ecology about how to maximize biodiversity, for example, through the intermediate disturbance hypothesis. And finally, you have ideas about how spatial structure can be relevant to the accumulation of complexity in ecosystems, where you have meta-ecosystems and diversity among them, and migration between them that allows for recombination and the formation of new ecological states. So in an origins of life context, we often think about these chemical ecosystems as persisting in spatially structured environment like adsorbative mineral surfaces, moving across those surfaces where they're effectively concentrated, where their diffusion is effectively lowered. And those different ecosystems might interact. When they interact, they might annihilate, they might continue to coexist, they might fuse and modify one another. And so we wanted to understand how these chemical ecosystems behave in space. So the earlier kind of ecological time series that I showed you were all in well-mixed reactors. Now we're going to consider these systems in space. So here's an example where a cycle is seated in the center and diffuses outwards, kind of spreading to new sources of food. And of course, we know that spatial dynamics are important for autocatalytic chemical systems. We have classic examples like the Belousov-Zhabotinsky reaction, this out of equilibrium process. We know that they can form stable patterns. So I want to consider cases where cycles mutually inhibit one another, because getting more complex ecosystems is easy when there are mutualisms between the cycles. It's hard when you have inhibition between them. And there are various ways that cycles can inhibit one another. For example, you can have the waste of one cycle interfere with the food of another cycle, or you can have their member species annihilate in a much simpler example that I show in the bottom. This is the one that I'm going to be working with in the subsequent slides. So you can put these in space. Here I have a hexagonal lattice with chemicals reacting within each site and diffusing between sites. And I'm coloring them according to the fraction of member species that belong to one of these two mutually inhibiting cycles, red for cycle A, blue for cycle B. And with slow diffusion, they stop interacting and just form these stable patches. With much higher diffusion, you can see one cycle starts to crowd the other out globally. And we can look at this more systematically, varying the diffusion of all the chemicals involved, kind of gradually increasing. In the low diffusion case, you end up with pretty randomly distributed ecological outcomes. In the very high case, you end up with one global winner, one cycle driving the other extinct. And you can look at the heterogeneity of chemicals across this hexagonal lattice. And what we find is that in the intermediate diffusion regimes, this heterogeneity is maximized. And insofar as these cycles are inhibiting one another through these reactions that might produce other chemicals that could help to provide support for the activation of future cycles, you don't have that sort of interaction in the very, very low diffusion case, and you also lose it in the high diffusion case when one cycle is driven extinct. And so this suggests that some intermediate diffusion regimes are the most favorable to kind of this biodiversity, so to speak, of these chemical ecosystems. That's all with keeping these cycles on an equal playing field where they have the same reaction kinetics and the same diffusion properties. What I want to show next is when you vary the properties of these two cycles asymmetrically. So I'm showing time series here on the right of both cycles being seated in some central site in a 3x3 array, and they can diffuse outwards. I make cycle A in red fiercer so that it consumes food more quickly than cycle B, and because of that, it starts to drive cycle B extinct initially. But cycle B is made faster than cycle A, meaning that it can diffuse outward more quickly and access new sources of food. And so despite initially being driven down, it manages to dominate in these outer sites and eventually reinvade, so that in the long term it ends up driving cycle A extinct. And you can kind of vary these properties systematically and construct a phase diagram. Here I vary the relative fierceness and relative fastness of the two cycles. And you find regimes in which either of the cycles can be favored, suggesting that spatial environments can select for new types of traits, such as the diffusivity of these member chemicals, once you put them in a spatial environment. A well-mixed reactor would be blind to these sorts of traits. Notably, you can also look at other types of inhibition that don't involve explicit chemistry. You can look at competition for adsorption sites on mineral surfaces, and there we find very similar dynamics. And I think that this is suggestive that something like the intermediate disturbance hypothesis might hold even for these chemical ecosystems. And of course, in an origins of life context, there are lots of different types of disturbances that you could have. The noise of the spatially structured environment that you have, different impacts. And so with that, I'd like to acknowledge David Baum, my undergraduate advisor at the University of Wisconsin-Madison, and Chris Kempis, my mentor and collaborator at the Santa Fe Institute, some excellent grad students and postdocs, including Prof, who just defended his thesis last week, and some excellent undergrads that I've had the opportunity to work with, including Gage, actually. He should be on here. All right, with that, I'll take any questions. If there's time. Okay.
Also, if everyone just wants to give their name and affiliation and ask a question, that'd be great.
Hi, my name's George Scheibel. I'm from Montana State University. And I should preface, I am a microbiologist. So, but that was a great talk. I thought you did a good job breaking it down for us. I'm just curious, when diffusion happens, I think of it in like three-dimensional space. So you were showing a lot of two-dimensional graphs. So what does that look like in three-dimensional space?
Yeah, so I didn't do simulations in three-dimensional space, in part because I think the most origins of life relevant spatial structures are going to be these two-dimensional environments. I think the two-dimensional environments can help to constrain kind of the effective diffusion that you would have. They can concentrate chemistry. Lots of origins of life theories that look at adsorptive mineral surfaces already, so we were kind of committed to that sort of scenario. And here, the idea of intermediate diffusion being favorable for chemical diversity, I think that's further support that's the type of scenario that might be important. I think there's a quote from Günter Wasserschauser, who kind of pioneered some of these ideas, that we don't build planes in the sky, we build them on the ground, and that's for a reason.
Just to follow up with that, what kind of mineral surface would you consider this happening?
Yeah, so kind of the experimental side of my group in undergrad, we were using pyrite surfaces, but here we're chemically agnostic, and we're also agnostic to the exact type of mineral surface.
Hi, I'm Ellie. I'm from CU Boulder. And I was wondering, so I'm mainly a lab chemist, and I was curious if you had any personal thoughts of how you would want a chemist to do some of this work, or what kind of questions you would want a lab chemist to do to help ground truth some of the modeling work that you do. Because I work a lot of mineral absorption and mineral facilitated chemistry, and of course when we do a lot of this stuff, it is an equilibrium reaction, because you're kind of waiting for it to resolve to equilibrium. We're basing our reactions on that. So how would you kind of design something like this, or theorize, or hope for something?
Right. So a lot of what I showed, that in the well-mixed cases, we were working with chemostats, and certainly chemostats are an experimentally kind of realizable approach. There's another side to our group that looked for kind of real autocatalytic cycles and known chemical reaction networks, both biotic and abiotic. And so I think one promising approach is to look at how these chemical ecosystems might explore that space where you seed with one chemical that activates maybe a single cycle or a small set of cycles, and then through the addition of subsequent seeds, you might activate kind of new cycles. And so I think there are kind of experimental approaches that can be done there, and I think people in our group are continuing that.
Okay, hello. We have a question from online, actually. So this is, comes from user Alex. He's asking, can you simulate tipping points hysteresis spatially with this method?
Tipping points. It's hard to ask for a clarification via Zoom.
Okay, Alex, if you can hear this, please clarify your question, and we'll come back to you. Thanks.
Okay.
Hi, I'm Jake from UCSD. Can you clarify why you used a stochastic process instead of like just solving differential equations analytically?
Yeah, so you can have cases in these chemical ecosystems where there's kind of some unstable fixed point. And those unstable fixed points, you need some degree of stochasticity to break out of them. So that's one reason. If I just use deterministic simulations, you could get stuck in those. It's also the case that in ecology, we're interested in lots of contingency. And stochasticity and then doing lots of replicates allows you to explore all those contingent outcomes. That's largely the reason.
Okay, we have time for one more question.
Hi, I'm Jolla from Indiana University. I'm an ecologist by training, and it was really interesting to see all of those theories that I've been trained for in this context. As ecologists, we abandoned intermediate disturbance hypothesis because we haven't observed in nature for 20 years. I was wondering if there are ways to test experimentally. What do you think about that? And also a clarification for tipping points. I think the online person mentioned you can probably see the signals before transitions, like increase in time autocorrelation or coefficient of variation. And so on.
Yeah, absolutely. Thank you for the question. Yeah, so I think in ecology, the intermediate disturbance hypothesis is controversial, and many people have abandoned it. I think in this kind of modeling context, it's very easy to kind of look for it, just because we're doing experiments in silico. And I think that if it does hold in the biological case, certainly it holds in the chemical case. There's a possibility that it holds in the chemical case and not the biological case. I think the weak version of this is that there are certain types of disturbance regimes that can be beneficial for chemical diversity, even if the intermediate disturbance hypothesis doesn't hold writ large. To the tipping points question, I think you do see sort of kind of signs. In the diffusion cases, I kind of vary it. You end up seeing sort of power law scaling in the patch size, sort of like you would see in an icing model in physics. And so the sort of theory that looks at tipping points there would also apply here.
Thank you very much, Alex. Okay, and our next speaker is going to be Mate Sarka from Atomkey.
Well, hello everyone. First of all, I'd like to thank for the organizers for having me here. And I have already experienced how great this community can be. I can tell that this is really a space for all. How many of you like chemistry here in the audience? Quite a few people here. So I will basically follow the logic of this quote throughout my whole presentation. So probably what I am doing and what we were doing is wrong, but some of it might be useful in the future. So amino acids. Amino acids, as we know, basically looking for amino acids is one of the key target for future in situ life detection missions. I'm going to talk more about the practical side of astrobiology rather than the theory that we have heard before. Maybe we can get together after it and find some new solutions for the problems and situations that we've encountered during our experiments. So there are three levels that we have to look at when dealing with amino acids. The first is kind of a qualitative problem. What type of amino acids can we detect and what do those tell us? And the second is the abundance of these amino acids. So their ratios, are they more similar to what things that we see here on Earth as the metabolic processes of microbes, or something completely different? And the third is basically what makes it a lot more interesting, or puts the icing on the cake, is the chirality. As you well know, amino acids are basically chiral, meaning that they have two versions of the same molecule, and they're non-superimposable. Basically, they're mirror images of each other. They have exactly the same amount of atoms, bonds, but they have a totally different structure when we're talking about the isomers. And getting to know the ratio of these amino acids is basically like a smoking gun evidence for life, if you like. But what does it have to do with radiation? Well, as you all know, as we sit here, we are experiencing the effects of radiation. But in the early universe, the activity might have been a lot bigger than it is today. And there are all sorts of theories that are, some of them are quite established, some of them are in the process of proving. But the point is that all the suns and stars basically produce ultraviolet light. And if you have this scattered from the surface, basically you have a polarized light. And basically, amino acids and enantiomers, which absorb the specific light better than their counterparts, then basically they get destroyed in the process. This is the same way actually as we detect their ratio, but it's also their doom if we are talking about high intensities. So this is a pretty much established model. You can basically replicate these using all sorts of accelerators and all sorts of sources to basically mimic this effect. The other one is more of a, still about photons, but more of a having the magnetic effect also taken into account when dealing with how radiation affects these molecules. And the third is basically stepping towards particles and subatomic particles, and how they change the isotopic ratio, and through the isotopic ratio, how they change basically the composition of the amino acids, and having them change their chirality. So these are just the main theories that we know so far that work. And in our laboratories, we basically set out to mimic some of these effects. At the accelerators at Atomcu, we have 3 of them, a small, a middle one, and a big one. They are all considered small in the field of nuclear physics. But for our experiments, we basically used a tandem accelerator, which allows a pretty quick change of ion sources. So basically, if you want to irradiate your sample with hydrogen, and then you want to see how the same sample is affected by a heavier ion, basically you can change the ions in a matter of hours, or even less if you're experienced. So it has a lot of versatility. That's the point of having a tandem accelerator. So on the image, you see probably the closest thing to a real lightsaber. And what you see here on the image in the top right is basically how hydrogen ions look like when they're extracted to air from vacuum. And basically, this shows the ionization of the air that the accelerated ions are hitting. So I'm going to jump a bit to chemistry, and you will connect it later. But the point is that we use the technique of capillary electrophoresis to basically see what radiation might have to do with the molecules and their chirality. CE is really a very, very friendly technique, and it's very, very simple. That's what people are basically saying who are expert in the field. But the point is that you have a tiny capillary, a tiny fused silica capillary with an inner diameter, let's say 50 microns, and an outer diameter of, let's say, 400 microns. And when you fill this capillary with an electrolyte and you apply voltage on this system, basically you will see stuff migrate, and they will migrate according to their hydrodynamic volume to charge ratio in this field. So what it allows you to do, it allows you to separate molecules based on their hydrodynamic volume to charge ratio. And if you put a detector in at a specific point of the capillary, basically you can see the molecules migrating through the capillary, and the specific point you get intensity versus time. So you see migrating peaks. This simulation on the left would like to show that, but unfortunately it doesn't run, so you have to believe me that the peaks you're seeing are actually migrating through the detector window that we're looking at. And it is using laser-induced fluorescence. Well, why are we using laser-induced fluorescence? The main reason is that to achieve high sensitivity. You can imagine it like being in a dark room, sleeping, and you basically just open up or open your phone, and you have a lot of bright light coming in, even though if you're doing it in broad daylight, probably you're not affected that much. It allows you to remove background, basically have great big signal. And to do that, you need some molecules that basically have a fluorescent property, meaning that you excited them with a specific wavelength, and they respond to you with a different wavelength. This is a molecule that we used to do the conjugation of our amino acids to basically enhance the selectivity and enhance separation efficiency. So in order to do CE, what you have to do is you have to have molecules that have a net charge other than 0, and we have to make them visible. This molecule here does the two things at the same time. So basically, what you see here is the succinimide from the dye reacting with the amine, forming a stable amide conjugate. And this way, basically, if you separate them, you're getting a pretty huge resolution. So what we did is basically first develop the buffer system or a background electrolyte, the thing that you fill the capillary with to do this separation. It's a pretty simple buffer. It contains 2 components. And the reason behind it was that we had to keep in mind the restrictions that a possible future in situ life detection mission would have. That you're not allowed to have a ton of regions, you're not allowed to have all sorts of mixing to happen. You have to make everything simple. That was the mindset that we had when we were doing the experiments. And as you see, we managed to separate actually 15 amino acids chirally of the 17 that we had in mind. Why is it a promising technique? Just like I said, a few moving parts, no power consumption, easy to implement, and it's just basically very, very friendly technique. And guys at JPL basically are developing this kind of technology. And what you see here on the image is basically the base plate of this whole setup is basically the size of your laptop. Actually, what we did during the irradiations, well, we made some sample holders from a drill press, a kind of a modified drill press, and we made 100 micron thick pellets of racemic alanine, and we irradiated them. Here you see the simulation how the proton beams would actually behave in a 100 micron thick alanine pellet. And as you see, towards the end, around 80 microns, all the ions stop. And this is actually where the most interesting things happen during an ion beam analysis or irradiation as well, which you see here a bit in more detail. So we have the protons coming in the vacuum from the accelerator, and then we have a window where the protons are extracted to air, and then as they enter the amino acids, they basically lose energy. And the nice thing about the ions is that they give off almost all the energies right before they stop. And this is actually why it's really, really useful during proton therapy. So we irradiated these samples with these energies and we designed a system to basically stop all the ions in the sample and see how destruction or any alterations occur. So moving forward, basically, you see that this is an expected electropherogram of a DNL, racemic amino acid. Some of you might see that these are not exactly the same height and not exactly the same area. This is just the control that we were using, and we compared all our results to these control runs. So basically, we have two big peaks, and we are happy. And if we go further, we'll be interested more in the small peak part of the electropherograms as we go with the irradiation and as we go as a function of those increases. So what you have here is basically the control that I just showed and different doses that have reached the sample. And it's apparent that some of the peaks are clearly increasing. Some of them show nice correlation. And some of these peaks are basically coming in duplets. And this is what made us think that actually, although we were irradiating racemic alanine, we are basically seeing racemic products. We're still in the process of identifying these molecules and these radicals that we found, but they seem to correlate well with the dose. And also, another interesting thing happened when we were looking at the big peaks of the amino acids, and we were just wondering what could this mean. What you see in this picture is basically, as we go with the dose, the LD ratio is getting more and more hectic, which means that we're not seeing it going towards one enantiomer or the other, but kind of mixed, noisy version of the two as we go with the dose higher and higher. So in summary, basically, we put together a simple setup and measured some radicals that could form due to irradiation, and we've done it in a chiral manner to basically see whether our method is capable of separating these chiral amino acids, and more importantly, if we think about the next possible or the most possible places in our solar system where life could be, actually they are in a huge radiation environment. So I think it is really necessary to basically create a library of radicals simulated here in the labs to basically make the work easier and unload this from the scientists who will have to basically figure out how molecules are formed in these high radiation environments and what they are seeing on the results when these instruments send back the data. So basically, we're trying to establish a big library where all sorts of amino acids, mixtures and single ones, are basically irradiated with all sorts of radiation sources, and see what products are there and what can be identified using this technology. So I'd like to thank you very much for your attention.
Okay, we have time for 2 questions for Monte. Chad.
Hi, Chad Pozoriski from Georgia Tech. I was really glad to see CELIF because it's a method that I work with as well, and so seeing it being used around is wonderful. And I have a question about your chiral method, which seemed to work very well. Did you develop this in its entirety, or when did you develop it? Basically, I want to know all about the chiral method. Is it published? And yeah.
It's not published yet, but we've developed it in collaboration with JPL. Basically, the key thing here is that to do chiral separation, the way you can imagine is that when you have these molecules in a solution and as they migrate through the capillary, if you have some additional additives in your buffer, basically you can emphasize or basically manipulate how these molecules behave. Which I mean that if you have sugars like cyclodextrins, these are circular long oligomers. Basically, these amino acids are, as they migrate, they meet with these cavities, and they basically form an inclusion complex. They go in, they go out, they go in, they go out. And some of the enantiomers are staying longer in this cavity, and some of them are staying for a short period of time. And as they migrate, they basically separate. And this is what we see. And in this context, to basically answer your question, we had a lot of constraints. First of all, we are not allowed to have a ton of additives, a ton of stuff in the buffer, because an instrument has to be able to do this on its own. So we had two components in it, which is the HAPIS or HIPIS, I don't know how they say it in English properly, AGPES, and the other one is the MD40, which is basically similar to cyclodextrins, but it consists of sugar oligomers, or actually monomers. And as you go, you have at the beginning one sugar, one glucose unit, and as you go 2, 3, 4, etc. And these, after around 7, they start to become helical, and you have longer chains of these sugars, and basically it enhanced the effectiveness of this cavity movement.
Thanks. Okay, last question.
Hi, my name is Shiva Agrawal from Western Michigan University. So you have used protons for irradiation. Have you considered using leptons?
Not yet. We know it should be done and it should be interesting, but we wanted to use the facility that we have available. And currently, we are only able to do protons and heavy ions. It would be a nice thing to move forward and do all sorts of experiment on this field as well. I'm happy to collaborate.
Second thing is, have you used variable energies for protons? At what energies are you?
In this experiment, we used only one energy. We didn't want to have too many variables. It was hard enough to figure out whether, with a 100 micron thickness of a pellet, are we able to robustly do experiments. And fortunately, with a simple system and with great care, we were able to basically make it work. So this was the first goal that we wanted to achieve. And of course, we have all sorts of experiments either already running or in plan to vary energies, vary actually the dose rate, which is actually a much more important thing. It looks like it is a key factor in some instances. And also doing everything in vacuum, doing everything. It was in air, but we also would like to do it in vacuum and also in cold temperatures as well.
Thanks a lot.
All right, thank you very much, Mate.
Thanks very much.
Okay, and for our next speaker, we have Byung-Hwa Kang from Pohang University.
Thank you.
So feel free to go to Surfside, get coffee, tea, snacks, whatever you need. We also have that courtyard space. You can hang out here in the lawn. There's also another lawn over there. But yeah, we'll meet back at 10:30. Oh, and then also outside, there's all those coolers. Those are just full of seltzers, sodas, so just take whatever you want.
everybody. We're going to start the next talk session, Life on the Edge, and our first presenter is Lauren Lowe from the University of New South Wales.
Hi everyone, so I'm Lauren and I'm from Anna Lang's lab at UNSW Sydney in Australia, and today I'm going to tell you a bit about the work I do on model protocells or model primitive cells. So I'm sure many of us here are trying to answer the question of how did life on Earth begin? Now I'm focusing on a very, very specific point in time when life had maybe begun to transition from super simple primitive cells, perhaps composed of a fatty acid bilayer membrane, to more complex protocells or primitive cells, more akin to modern biology. At this point in time, we'd started to see the emergence of phospholipids and some simple enzymes, but we hadn't yet seen the emergence of all the complex cellular machinery that exists in our cells. So things like membrane transport mechanisms, for example, that transport nutrients and waste molecules out of the cell, maybe they hadn't come around yet. So that begs the question, how are these protocells actually able to feed themselves? How were they able to grow? How were they able to divide? So I'm trying to answer this question by building a propagating synthetic cell. What that basically means is it's an artificial cell that is able to feed itself, it can grow, it can divide, and I'm trying to achieve this using the simplest components possible to try and understand how life transitioned into what we know it as today. So this involves a pretty large collaboration, and with my collaborator in Japan, Yutetsu, he's been able to build a preliminary synthetic cell composed of a phospholipid bilayer membrane, also known as a vesicle. And this vesicle is composed of a mixture of POPC and POPG, two types of phospholipids. And within this vesicle, we have a system that can be used to actually synthesize fatty acids and eventually synthesize phospholipids that can then be incorporated into the bilayer membrane, actually allowing it to grow. Now, one of the big problems with this system is that you can only encapsulate a small number of nutrients actually within the synthetic cell, within the vesicle. There's only room for so many molecules. And if you encapsulate as many as you can, you only really get 1 to 2% membrane growth, which isn't really sustainable for a cell that we want to make grow and divide. So what we really need is some sort of regular nutrient supply. So we need an external feedstock of nutrients that can actually permeate the lipid bilayer membrane, so make its way from the outside of the cell to the inside of the cell. That way we can actually achieve continual membrane growth. So that's my role in this project. I'm trying to figure out a way that we can actually get the nutrients that we need for the synthetic cell to function to make its way across the lipid bilayer membrane to the interior of the synthetic cell. So in order to do that, I've needed a technique that can be used to monitor solute permeability, or the permeability of different nutrients. And to do that, I'm using what's known as a shrink swell assay. Basically, that involves preparing vesicles encapsulating a fluorescent dye known as calcine. When you mix these vesicles with your solute or your nutrient of interest, the difference in osmolarity actually causes the vesicles to shrink because water rapidly exits the vesicle, and we kind of see these shriveled up, sort of shrunken structures. And because we've had water exit the vesicles, we've actually had an increase in concentration of the dye that's on the inside of these vesicles, and that actually causes the fluorescent signal to decrease because calcine is a self-quenching dye above certain concentrations. So at high concentrations, the fluorescence actually goes down rather than going up, so it's a little bit counterintuitive. But over time, if we have a permeable solute, so a solute that does actually enter the vesicle, we'll start to see the vesicle slowly swell up again, and it'll roughly get back to its original size. And with that, we have a decrease in calcium concentration, so we have an increase in the fluorescence, roughly to what it was to begin with. And we can basically monitor that whole process using some sort of fluorimeter or something that can measure fluorescence, and we can monitor the changes in intensity over time. So we can see initially the intensity is somewhere up here, and then with the addition of the solute, the intensity drops down, and then if we have a permeable solute, that signal will start to recover, and we start to see the original intensity kind of values there. Now, because these intensities are kind of a direct measure of the calcine concentration, we can convert concentration to volume. So the plots I'll be showing you today show the changes in volume over time. And it's just worth noting that the initial stage where the vesicles shrink and shrivel up actually happens very quickly, and the experimental setup I currently have, I can't actually see that because it just happens too fast. So the data I'll be showing you is just the recovery of that volume. So the basic workflow for a lot of my experiments is to first prepare the vesicles composed of an equal mixture of POPC, POPG in 50 millimolar HEPs, so that's just the buffer. It's roughly buffering around physiological pH. And these vesicles all encapsulate that fluorescent dye known as calcine. I then make sure all the unencapsulated calcine is removed, and I mix the vesicles with the solute or the nutrient of interest and monitor the changes in fluorescence over time, and then determine the changes in volume over time. So this is what some of the data looks like. On the left, we can see some examples of some permeable solutes. So our control here was the 50 millimolar HEPs in the blue, and all of this data is normalized to the volume of that control at time 0. So we see in the blue, the 50 millimolar HEPs, it's just a flat line there, not much is changing, and that's what we'd expect. It's our control, it's what we made our vesicles in, so we don't expect any volume change there. But when we mix our vesicles with something like glucose or glycine, for example, so the red and the purple, we see that characteristic curve telling us that the volume is actually increasing after the vesicles initially kind of shriveled up. So that tells us that these two solutes are both permeable across these lipid bilayer membranes, and glycine is actually more permeable than the glucose because it increases at a faster rate. On that plot, I also have glycerol. At first glance, it kind of looks like nothing happens there, but glycerol is known to be super permeable across lipid bilayers, so it's likely that I just actually missed that initial shrinkage and swelling stage, so it just looks like nothing's happened right now because I missed the first kind of couple minutes of this process. Now on the other plot, we have some examples of some impermeable solutes, and you can see there's quite a few there. So a lot of the solutes I've looked at so far are quite impermeable, and that's we can see that there's like those flat lines at the bottom there. There's no increase in volume over time, so over this time frame, none of these solutes are permeable, which is kind of disappointing because a lot of these solutes would be useful nutrients for a synthetic cell. Things like AMP, ATP for energy, and then something like sodium acetate I was hoping to use as a carbon source to actually synthesize fatty acids and then the phospholipids. But it's not particularly surprising that a lot of these solutes were impermeable. It's relatively well established in literature that phospholipid bilayers aren't the most permeable things, especially when comparing them to a fatty acid bilayer, for example. So fatty acid bilayers are kind of useful to think about because they have been proposed to have made up the first protocell membranes before we saw the emergence of phospholipids. And we can see in this plot here for something like glucose, it's a bit less permeable in phospholipids than in fatty acid bilayers. For something like sodium potassium, it's significantly less permeable, and like over this scale, that's a really significant margin there. And then for something like AMP and magnesium, that's not actually on the scale shown here for phospholipids, so it's way less permeable in phospholipid membranes than it is in fatty acid membranes. So again, these results aren't particularly surprising, but I still wanted to try and find a way to actually get these nutrients to permeate the bilayer. So today I'm going to talk about a couple of different strategies I've tried to use to actually improve permeability, the first of which is modifying the membrane composition. So as I've said, all the vesicles I've been working with so far have been composed of POPC and POPG. Now these lipids are quite nice to work with. They're cylindrical in shape, so they pack together in these really nice bilayers. They're very uniform. But what would happen if I were to, say, disrupt the way the lipid molecules actually pack together? What if I introduced a lipid with a different shape? Something like lyso-PC, which has the single carbon chain rather than two, but it still has a relatively bulky head group, so it has this more conical-like shape. And when that gets inserted into bilayers, it can cause defects to form and pores, which would potentially allow nutrients to pass through the membrane. So I wanted to explore what effect adding small quantities of lyso-PC would have on permeability. And the other lipid I looked at is oleic acid, which is a fatty acid, has a single carbon chain and a relatively small head group. But because it's a fatty acid, it has very different properties to what phospholipids do. So I wanted to explore how those different properties might affect the permeability of the membrane. And generally, I found that they didn't make much of a difference. So first of all, I prepared all the vesicles the same way I did before, but added about 10% either lyso-PC or oleic acid to the system. And generally, yeah, as I said, didn't make much of a difference. We have one of our example permeable solutes there, so glucose, which we saw was permeable earlier. We can see in those three curves there isn't really much difference between them. If anything, the green curve, the oleic acid, has shifted down slightly, but the shapes of the curves are all very similar, which is telling us that the permeability hasn't really changed. And we see something similar with one of the impermeable solutes, that being lysine. We still have all those flat lines, so we saw no improvement in permeability there. And this was generally the case across a lot of the different solutes or nutrients that I actually tried. There weren't any marked improvements in permeability, except for when I tried sodium acetate. So when we had the presence of 10% oleic acid in the system, we see a significant improvement in permeability compared to what it was originally with the POPC-POPG system, and also compared to adding the 10% lyso-PC. So we do see that curve that's telling us that the vesicle is swelling up again, and we actually have the solute molecules permeating into the vesicle, which is good news. We saw that one of these solutes actually kind of worked and actually were able to permeate the membrane. And as I said, it was great to see that it did work with sodium acetate because we wanted to use that as our carbon source for fatty acid synthesis, then phospholipid synthesis, and that would actually help getting the membrane to grow. But as I said, overall it didn't work for a lot of the stuff I tried, so I wanted to try a second strategy to try and improve permeability, and that was through the addition of divalent cations. So in literature, it's pretty well established that divalent cations also affect the packing of the way of the lipid molecules, so the way the bilayers actually pack together. So I wanted to see if that would make a difference to permeability. And again, generally, I found it didn't make that much of a difference, except with the presence of sodium acetate. So with the 10% oleic acid system and the sodium acetate, when I added magnesium chloride, which is what we see in the orange, there was an improvement in permeability. We see that curve kind of increases faster than what the blue line, which is just the pure sodium acetate, and the green line, which is just the magnesium chloride. So for whatever reason, the addition of magnesium chloride with the sodium acetate and in the 10% oleic acid system did improve permeability overall. But again, generally, I found that this was quite difficult to achieve in a lot of the solutes I tried. I've also tried other divalent cations, which I haven't shown here. In general, it was pretty tough. So what does that mean overall? So generally, I found that permeability can somewhat be modulated for specific solutes by changing the membrane composition, and divalent cations can somewhat improve the permeability of specific solutes. But overall, it was pretty difficult to actually see any marked improvements in permeability for a lot of the different things I studied. So what does that mean for origins of life? Well, perhaps we needed protein channels to actually co-evolve with phospholipid synthesis. As I mentioned earlier, fatty acid membranes are much more permeable than phospholipid membranes. So if they were the first protocell membranes, they were able to access nutrients a lot easier than phospholipid membranes on their own would be able to. So we probably needed some sort of mechanism to actually co-evolve with phospholipids as they started to emerge. But this is very much an ongoing project. I'm trying a lot of different membrane compositions, a lot of different divalent cations, and I'm still working on other methods to kind of improve permeability. So hopefully in the future, I'll have some more definitive results with some improvements in permeability for a lot of other solutes too. So with that, I'd like to acknowledge the entire WAN group and my supervisor, Anna, for all their support. Thank you to my sources of funding, and thank you to everyone for listening.
Okay, we have time for a few questions.
Thank you. Hi, I'm Bolaga from Ohio University. I study molecular biology. I'm just interested in the method that you use to measure your membrane permeability. Is it patch clamp electrophysiology or what?
So the technique I was using is known as a calcine shrink swell assay. Basically, it's not a direct measure of permeability, it's actually a measure of the changes in volume, but it's reliant on the fact that with those changes in volume, they can only occur because the solute is actually permeating the membrane. So it's actually a fluorescence-based technique, so I use a fluorimeter or a plate reader, depending on the experimental setup I have going, to actually measure those changes in fluorescence over time. And from that, you can calculate the changes in volume, which you can actually then go on to work out permeability coefficients from those curves, which is something I haven't done yet, but you can actually fit a model to that curve and extract the permeability coefficients as well.
Okay, I'm just actually thinking that if you can employ the patch clamp electrophysiology method, it can give insight into like membrane polarity, hyperpolarization, which can also give more insight on this.
Yeah, 100%. And there's a few other techniques that I've actually used as well that are quite similar to that, but I didn't show them in this presentation, but I'm happy to chat about them more after if you're interested.
Hi, I'm Tim from University of Wisconsin-Madison. I was wondering, do you think there could be an effect of vesicle size, or specifically surface area, on the degree of permeability? And if so, did you like control for size? Did you extrude your vesicles somehow?
I sure did. Good question. Yeah, so these vesicles for this system I'm working with have all been extruded through 50 nanometer pores. They're roughly, they're probably about 40 nanometers in diameter. For the most part, it's a relatively even distribution. I did do DLS on pretty similar systems. Yeah, but that's why I did extrude, because I would expect permeability to be affected by the size of the vesicles.
Thank you very much, Lauren.
Thank you.
Okay. Our next speakers will be Jonah Abashir from Portland State University.
Hello, everyone. My name is John Abshire. I am a second-year PhD student at Portland State University, working in the Center for Life in Extreme Environments, more specifically out of the Extreme Virus Lab. And today I'll be giving you a little bit of insight into life in hell, understanding the role of toxin-antitoxin systems in prokaryotic genomes and their potential for virus-host coevolution, TLDR, extreme organisms, the viruses that infect them, and their interactions in these extreme environments. And our journey kind of begins right here. So this is a personal image of Lassen Volcanic National Park. It is one of the many areas on our beautiful planet that is home to these volcanic hot springs. This is one of the main sampling sites of our lab. And while most people wouldn't believe that there are things living in this environment, there certainly are. Specifically these microbes from hell. And I'll get back to these guys in just a second. But a little bit of background about why we study these particular environments. So these volcanic hot springs are often considered and understood to be analogs for planets both outside and inside of our solar system, referring back to some of the ancient hot springs found recently on Mars, which may or may not have looked something like this boiling mud pot that we find in Devil's Kitchen at Lassen Volcanic National Park. Getting back to the microbes, one such microbe is this microbe right here, spherical cell known as Saccharolobus sulfactericus. This is a scanning electron microscope showing you the topology of the cells. Coined S441 by our lab, really thrives and lives in these hot acidic conditions with temperatures from around 75 to 80 degrees C and pHs of around 3 to as low as 1. So these really extreme environments. And when we think about these environments and the rich ecosystems that habitat them, we can also think about one of the most abundant molecules on the planet, so that of viruses. And yes, there are these extreme viruses infecting these extremophiles. The one which I work with in lab is Sulfurobus spindle-shaped virus 1. Saccharolobus is a natural host of this particular virus, this SSV1. This is Stanley, uses they, them pronouns, really characterized by his lemon shape. So these shapes are really unique to both archaeal organisms, extremophiles, and some of these extreme viruses. And when we think about looking at these organisms and the interactions between them in these extreme environments, we can look at some systems currently prevalent today in bacterial cells. So these toxin-antitoxin systems, usually known as addiction modules, found on plasmids, again, really prevalent in bacterial genomes, only recently being discovered to have some viral encoding. And what this particular system confers is when you have a plasmid that has the positive, or is positive for that particular addiction module, that cell needs that plasmid in order to continue its life in that particular environment. So here we have the presence of the TA. Should that cell or progeny continue with the presence of that plasmid, you would expect normal growth in the environment, whereas if it were to lose that plasmid, it would have been addicted to that particular genome, it would die off, whereas in a negative system, you would get growth either way, whether or not there's plasmid loss. And we can further visualize this particular mechanism by looking at what might happen to some uncolonized cells. Some sort of event happens in which the addiction module is introduced to that population, and you have your addicted survivor going on to make new progeny, and then conferring that group protection and persistence. So these systems are really understood in bacteria to confer a microbial persistence phenotype. Some have been referred to as phage defense mechanisms and largely considered as plasmid stabilizers on the plasmid. And we can also assume that another group of uncolonized cells were to come in here, perhaps that group encounter, you would still get that toxic culling from that toxin-antitoxin system. One important note, they are characterized as two genes that are typically right next to each other. The toxin being just downstream of the antitoxin, pretty stable toxin, pretty unstable antitoxin. And looking at this system with SSV1, a previous student did quite a bit of work in mutagenesis of the SSV1 genome. And we use this, at least these mutants, to test whether or not some of these genes or open reading frames are essential to the virus. And two genes in particular, this T3 and TX transcript, these particular two genes, we can make changes to what I think is the antitoxin, and we don't see too much difference in terms of viral infection, viral function, whereas if we were to delete this or insert a sequence into this particular open reading frame, what I think is the toxin, we do see differences in infection mechanisms. And looking at infection mechanisms of these extreme viruses and just virus reproduction overall, viruses can typically go through two pathways or a combination of both. So a lytic cycle in which the viral genome, the viral DNA will insert itself into the cell. You have some replication going on, eventually assembly of those virions, and the cell will then burst, die, and those viruses will go on to infect other cells. Whereas in a lysogenic cycle, the viral genome is incorporated into the host genome, enters usually a latent phase in which it kind of just stays there, maybe some induction event happens. In SSV's case, it actually just buds from the cell without killing the cell. And one of the two ways that we use to test for this viral reproduction is, of course, PCR, so amplifying a sequence that's specific to viral DNA, looking for its presence in cell-free supernatants. Another way is through halo assay, so very similar to a plaque assay, in which you look where cells are bursting. We cultivate a lawn of cells, uninfected saccharolobus cells, like you saw on the slide a while back. And then we spot cell-free supernatants, which contain these mutant viruses, onto the plate and look for clearings. And this halo assay is made possible because of the fact that SSV1 buds from its cell without killing the cell. And the infected cells grow quite slower, so it really confers this growth stunting phenotype in the saccharolobus cell. In looking at the toxin protein, and a large majority of my work has been looking at mutants that we've made in this toxin protein, and several machine learning softwares have indicated that there's quite a high probability of a cleavage site over here behind this very hydrophobic helice right here. And so I sought to characterize at least this particular mechanism in SSV1 through using some of these mutants that we already have. And while largely these over here are wild type, so really shouldn't expect any kind of change in viral function, you can see some really nice halos, and we can confer that the cells are not dying, but they're sick. And in our mutants where we have insertions in that specific toxin open reading frame, we don't see any halo formation. Likewise, I sought to substitute those two residues at the cleavage site, since it's largely important for protein maturation, protein function, and we still don't see a halo just switching those two residues. But the main takeaway from this is that we still get virus replication. We still get quite a bit of at least high levels of viral replication from some of these mutants. And one of these mutants in particular, we didn't see anything, so maybe that one's detrimental to that particular open reading frame. But we do see that there is virus replication happening in the cell supernatant when we screen for DNA. And it's important to kind of note that this protein isn't just unique to SSV1. In looking at these systems in these extreme organisms, this one in particular up here is another SSV, so another spindle-shaped virus, in which we see really low sequence similarity across these particular genes, but quite a high bit of structural similarity between these genes. And a lot of these are genomes of extremophiles, while some are virally encoded. And one of the main takeaways, really just wrapping it all back around, looking for these particular mechanisms and how they confer persistence with both their extremophilic organism and their viruses would be a really nice first start in looking for these biosignatures, perhaps, and ways that we can look into how the microbes might be interacting in these particular extreme environments. This is another sampling video just of Lassen Volcanic Park, pretty close to where S441 was isolated from. Looks pretty similar to maybe something we'd see on another planet that we could sample at. And then shameless plug, but looking for viruses in space, I think, is the obvious next step. We know that they're the most abundant biomolecule on the planet, and just understanding that these mechanisms and that these systems are out there allowing these cells to thrive in this particular environment would be a great starter conversation for looking for these small biomolecules in space. And with that, I just want to thank my lab, my group over here at Boiling Springs Lake when we collected samples, and my PI, Dr. Ken Stedman. Thank you.
Thank you, Jonah. We have time for about 3 questions.
Hey, my name's Pia, and I'm really interested in like CRISPR-Cas immunity of microbes. Have you looked at all into the interactions of other phage defense systems?
Yeah, so at least in terms of this toxin-antitoxin system, they're pretty understood in bacterial cells to like incorporate themselves into the genome as CRISPR-Cas9. There are some proteins that that look pretty similar to this A291 and some of these other virally encoded genes that do have some kind of CRISPR-Cas editing mechanism. So yeah, I think it might be leaning towards that way, but they're pretty well characterized in terms of like how they work with proteins and mRNA. So yeah, not too far there yet, but definitely somewhere in there.
Thank you for the talk. Marshall Seaton, JPL. And I was curious, so you're talking about looking for viruses in extraterrestrial environments. Do you think there would be, or I'm curious, I'm completely ignorant
yeah,
on the subject.
No
I dropped
worries.
out of bio too. So for this, for viruses specifically, like would small molecule biosignature classes vary from viruses to like what you'd be looking for with cells?
Yeah, yeah, I for sure. And that kind of brings me at least back to a point like of looking for fossils and stuff like that. So viruses in sediment. And at least our lab, there's been some work in like silica encoding viruses and how, you know, the stability of them. So maybe there's a pretty silica-rich environment in which we might be able to look for any kind of these signatures. So like proteins or yeah, just specific things that might have been preserved in those particular environments. Considering that these viruses live and thrive in pretty extremes, you know, they got to be out there, at least in my mind.
And so you mentioned looking at like morphology and fossils and things like that. I know that at least for looking at cells and things, they've been shown to mimic abiotic systems pretty well because humans are very good at pattern recognition. And so has anything been seen like that for viruses? If you're mentioning fossils?
Yeah, not that I'm familiar with. I know it's at least a brand new conversation of starting to look for like viruses in space, astrovirology, things like that. So yeah, we're just kind of getting up and running.
Oh, yeah, no, sorry. I don't mean to like play 20 questions.
Oh, no.
I just thought it was
totally
cool.
fine. You're totally fine.
Thank you.
Yeah. There's a little cure lab that like goes on right now that I teach, so I'm obviously not there, but there in spirit.
Any other questions? Our next speaker is Fatima Liho from the Earth Life Science Institute.
Let me just set up my timer.
Okay. And for the last talk, we have Manette Pena from SOMA.
Hi, everyone. Thank you for having me. This is a very special moment for me because I am going to talk about hydrothermal vents. So being Scripps is a very special moment. So I'm happy to share it with you and also about my PhD journey. So the link in hydrothermal vents is very important between the ocean space sciences. Since its discovery in the 70s, the efforts to study the deep ocean and these extreme environments has increased a lot in these last years, also because of all the technology and all the improvements that it has been made through study the deep sea. And also, not only it was an outstanding discovery for geophysicists, geologists, also for biology and microbiologists, that we found these extreme ecosystems when we are finding these extremophiles that are high, with a high diversity, not only in microbiology, but also on macrobiology. And in a bigger scope, hydrothermal vents are very important for astrobiology. Bigger scope is the origin of life, which is a very important thing in our planet. And also, for looking life elsewhere, also hydrothermal vent systems has been proposed not only as place where these structures have created or originated the molecules for the cells, but also for probably there are similar structures existing in the ice-moons. So this is one of the biggest scope that we are studying hydrothermal vents. But in particular, in this topic, we are exploring what is the Gulf of California in Mexico. So if you're will hit the Gulf of California, we are here in Scripps at the north. If you cross the border, here Tijuana and Ensenada, excellent tacos. If you want to stay longer, you can tell me. But for studying the Gulf of California, the deep ocean has been studied for over a decade. And they have found that there is a series of transform faults and spreading centers, which have led to the discovery of hydrothermal vent systems. In particular, we have found, here it is, in Pescadero Basin at the south of the peninsula of the Gulf of California, we have found two hydrothermal vent systems. I'm sorry, they moved. But here, the yellow dot, it's called Auca, and the red dot, it's called Jaigma-Ja'a. Auca was discovered in 2015, and Jaigma were discovered in 2018. These are the hydrothermal vent fields that are at a depth of 3,650 meters depth. This is the deepest known hydrothermal vent system in the Pacific Ocean, which has led us to study them as a very extreme ecosystem. And we have found that the geochemistry of these hydrothermal vents is very unique. They are made of calcite, and there have been some temperature measurements between the 20 degrees to 100 degrees Celsius around the sediments, but one of the highest hydrothermal vents temperature goes around 300 degrees. So one of the questions that we have, one of the big questions that we have for studying these hydrothermal vent systems is who are the microbes related to the hydrothermal fluids in the sediments of the Auca and Jaigma hydrothermal vent fields, because we don't know a lot of these hydrothermal vent systems. So the first question was, who are them? So for this, an oceanographic expedition was made in 2018 aboard the research vessel Falcor. This research vessel has a ROV called Sebastian, which is the rover that let us submerge, and with a robotic arm, it was able to take some samples of the sediments. So here you can see a little insight about how are the sediments around these hydrothermal vents. All the things, all the white stuff that you see around is microbial mat. So we collected around 30 push core sediments, and all these core samples were sectioned on the ship And it was subsampling with, for taking DNA extraction to do pore water analysis. Back in the lab, we made some ion chromatography, DNA amplification of 16S, some sequencing and bioinformatics, and of course some statistical analysis. And so here I'm going to present you some of the results of this. So one of the questions we also have in the first place is how are AUCA and Jaigmaja compared? How are these hydrothermal vent systems related? Because AUCA and Jaigma are like 2 kilometers away. So we wanted to compare how much are they similar in terms of the geochemistry and all the microbiology that we have been finding. So we performed this MDS analysis. So if you see there, we have 3 different locations. AUCA and Jaigmaja are the hydrothermal vent fields, but we also have another location that is called Uwagdum. I'm sorry for the names. All the names are native words from the region. So that's how we decided, well, scientists decided to name them. These both are hydrothermal vent systems, but Uwagdum is a site between them, but it's a dead hydrothermal vent site, so it's not active anymore. So we use it as an outsider to do this test. So in general terms, you can see that they are very similar, AUCA and Jaigma. And here this is the outsider Uwagdum, that it looks not that similar to them. So they are sharing a lot of things, but we don't know what, if it's the geochemistry of this, the microbiology. So we want to go further in to study the microbial composition of both of them and compare them and to see if they are related or what are the differences between them. And one of the biggest surprises that we have is that the microbes are related to the hydrothermal fluids, but between AUCA and Jaigma, the microbes are different. We have different film composition, but the only film they share is thermatogota. So this is a very interesting thing to explore, that we are finding different microbial communities between those hydrothermal vents, but they are sharing one. So this is telling us about the thermophilic and the hydrothermal fluids, all the ambiental, all the conditions, the extreme conditions that are around, and also they tell us about all the geochemistry that is happening. It's telling us about it's very complex, I think, all the metabolism that is going on between these sediments around hydrothermal vents. So we are trying to explore furthermore about this group, the Thermatogota, that they are extremophiles. They are adapted more than 50 degrees or 80 degrees Celsius. So we are trying to understand if these are like the same families that they are sharing or not. That's one of the next steps for this. But also the implications for astrobiology in terms of hydrothermal vents is that we are still exploring a lot of findings of thermophilic microbial communities in these hydrothermal vent systems to try to understand the limits of life on our Earth. As it is one of the deepest points of the Pacific Ocean, it is telling us a lot of information about that. And also, another step for the investigation that is going on, it's applying it to the icy moons with habitability models, where we can try to add these ecological models, and now we have data to try to simulate this. We have geochemistry data, we have microbiology data, so we can try to approach ecological model to the icy moons. If someone in the icy moons wants to collaborate, please let me know. Well, the highlights of these presentations to take away, I want you to show you some of the hydrothermal vents, how the hydrothermal vent system looks like in Pescadero Basin. But this is the first insight into Jaigmaja, a hydrothermal vent field, and its microbiology. It's the first time that we have seen this. our findings provide valuable insight into the deep sea biosphere, and that we are learning that there are a lot of information that still need to be processed and to know and to discover. And we have found a diverse range of extremophiles in microbial communities. Right now, I'm just talking about the thermophiles, but we have methane oxidizers, anaerobic microbial organisms. So there are a lot of things going on in the depth. And well, knowing that the Pescadero Basin is the deepest hydrothermal ecosystem in the Pacific Ocean, well, it implies that we are still understanding the limits of life on Earth and beyond. So with this, I want to thank you. Please, if you want to know a little more about the Mexican Astrobiology Society, there is a link. And I am also from the Astrobiology Lab at UNAM, and we are happy to collaborate. Thank you.
Thank you for your talk. I was just curious, do you find many nanoprokaryotes, like nanoarchaea or like candidate phyla radiation bacteria, or do you just filter them out in your analyses somehow, or like are there any interesting patterns?
Well, right now, we are finding also archaea and bacteria, but that's like the farther that we have. Like now, there is a lot still of information to show, but there's a lot of things going on.
Yeah, I just remember like looking through various papers. There are a lot of them, a lot of those like nano-sized clades present in the hydrosomal vents.
Like nanoarchaea, or?
Yeah, like nanoarchaea or.
Yes, we have a nanoarchaea is around, and AbGradCon is also in there. Yes.
I guess, sorry, just kind of curious. I'm a physics kind of background person from UCSD, and I was wondering about temperature management. So, like, you know, above certain temperatures, like you're going to get denatured proteins and stuff like that. And I was wondering if there's extremophiles that do like that cool themselves somehow, or anything like that. Sorry, just totally ignorant.
Yes, actually, well, right now I'm talking again as with thermophiles, but actually here in the hydrothermal vents, well, not the hydrothermal vents, around the hydrothermal vents, all the sediments, there are different changes in temperature. So right now I am talking about the highest ones that we have measured, but if you go farther, the hydrothermal vents, it drops to 2 degrees centigrade. So we are studying, well, it's able to study different microbial communities adapted to high temperatures, but also to coldest places around.
Any other questions for a minute? Oh, Kelly?
Hey, really awesome talk. This is really cool. I worked with hydrothermal vents as an undergrad, so this is like really nostalgic for me. I'm curious if anyone in your group or anyone part of the cruise is doing, and maybe Karen Lloyd wouldn't like this, but is doing any culturing from these environments.
Yes.
Like specifically, I'm thinking at high pressure because these are some of the like deeper vents in the Pacific, or the deepest vents in the Pacific, I think you said. And so I'm just curious if anyone's doing culturing and if they're including pressure in that.
Yes, actually, one of the here in one of the collaborations that we have is in Caltech, in Victoria Orphan's lab. Yes, the group are making some culture experiments with some of the different sediment cores in different sites. So they are trying to do that, and they have also published one paper about culturing archaea, I think. So they are doing it, but. Of course, the challenging things about the pressure. So they have like this tank to try to approach that. Yes, they are trying to do cultural techniques for this.
Any questions from up top? Any questions here?
I thank you for the talk. I have a very trivial question. We've been having issues with sediment samples in our lab, freaking out how not to contaminate them, or not being sure if they are contaminated when we are taking the samples. I wonder how you control for that.
Yeah, that's a good question. So when we are working on the ship back in the lab, there is a lot of possibilities that it got contaminated. We're trying to be as quick as possible and in clean spaces, but we actually, where we are working with sediment cores, it's mud. So it gets really mud all around. We try to clean, but we just try to be as quick as possible. One of the things that we rely on is the bioinformatic analysis. So in the bioinformatic analysis, when we are processing all the 16S data, the sequences, we are filtering them. So we have like we have also for the 16S or the DNA extraction some blanks and some control samples. So we tried, like, if there is a sequence in the blank on the control, we delete them. So we try to filter and to make the cleanest sequence possible. But yes, of course there is some contamination, and we also, like, sometimes we see like, oh, that's not from the deep sea, but we try to see the sequences and try to clean them. That's why. Mm-hmm.
Any last questions? Okay, thank you very much, Manette.
Thank you.
Okay, so lunch is served in Surfside, and then we'll be back for the short talk session.
the University of Maryland.
All right, hello everyone, how's it going? My name's Sean. I'm from the University of Maryland, Baltimore County, and today we're going to be looking at xenoamino acids, a look into biochemistry as we don't know it. So I'm going to fly through a lot of this because it's only a 6-minute talk, but please feel free to come find me at my poster outside afterwards. So my project attempts to merge theory with experiment in order to test if an alternative amino acid set is capable of building protein structure. Now to do this, we need to select a candidate alphabet from plausible alternatives, and from there use our collaborators over at the Charles University in Prague to build oligopeptides. Now, I'm going to take a step back here and go over some very quick background. So all life on Earth since Luca has used the same set of 20 L-alpha amino acids to construct metabolism. Now, multiple disciplines agree that there were far more than 20 available for early evolution and the origins of life. And over the last decade or so, there's been theoretical work that identifies a simple but statistical profile that distinguishes life's amino acids from alternatives. And that is that the 20 amino acids used by life exhibit a non-random coverage of size and hydrophobicity, when we define coverage as the range of values in a set and with how evenly they distribute across that range. So what do I mean by that exactly? I want you to consider a set of 5 amino acids and pick any physicochemical descriptor. This could be size, this could be volume, log P, this could be charge, and you're going to measure them, rank order them, and plot them on that axis. From here, what we can do is we can get the intervals between each amino acid, and this ties into our idea of coverage, which breaks down into 2 components, which is range and evenness. So range is simply just the sum of these intervals between each amino acid, and evenness is the sample variance of these intervals. Now this ties back into what I said, the coded 20 amino acids are statistically non-random, and 1 in about 2.5 million alternative alphabets would have the same or better coverage in size and hydrophobicity. Now, why exactly are we looking at range and evenness? Well, range allows for a broader diversity of structures and functions, and evenness allows for the best approximation of any desired physical chemistry. So I like to think of it as trying to recreate a black and white painting using a defined set of tiles, and you need to pick which colors you want to use to try and get that painting as close as possible. Now, range is effectively the difference between having black and white tiles versus just different shades of gray, and evenness is among the black and white tiles having an even distribution going from black to white. Now, why exactly are we looking at log P in volume? Log P is just a measure of hydrophobicity, and hydrophobic collapse is crucial for protein folding. And for volume, volume determines the physical space that allows for protein folding. So now that we have this idea of coverage in our mind, I would like to very quickly go over our workflow. I'm going to skip essentially everything except that top right corner where all we're doing is we have a heuristic search protocol that searches a library of purchasable amino acids. And from there, we are in a constant feedback loop with our empiricist collaborators in attempting to eventually get a candidate alphabet. Now, I'm sure you are all dying to see what one of these alphabets look like, right? So here we go. This is our current alphabet. Now, big emphasis on current because of that iterative feedback loop. This is definitely subject to change and probably will change before we start constructing oligopeptides from a set. But here is our first set. This is simply just one example of around 10 to the 14 possible high coverage sets that we could pick from. So there are a lot of variations that we can change and play with the empiricists. Now, again, the goal here is to synthesize these oligopeptides from a completely non-canonical set, and that's hopefully what we'll be doing in a few weeks. Now, I would like to very quickly end on a deeper note of who cares? Why would it advance science to design an alternative amino acid set? And I would like to kind of bring that question to this idea of, is there such a thing as a good or bad amino acid alphabet?
Now, before the empiricists in the crowd jump out of their chairs and strangle me, yes, the empiricists are saying, of course there are bad amino acid sets. The 20 are very good at what they do. But theorists are pushing back a little bit and saying, well, evolution can find any needle in any haystack. So I would like to believe that the truth lies somewhere in between, but what we're really trying to do here is begin to characterize good versus bad using an adaptation of 1980s behavioral thinking, so optimality theory, for life's amino acids. Now, I'd like to conclude just by thanking our sponsors, our funders, and our collaborators over at the Charles University in Prague. And if you would like to talk to me about this in much, much greater depth, please find me at poster 24, which is outside. Thank you all so much. I really appreciate it.
Our next speaker is Peter Winslow from the University of Minnesota.
So my talk is in a similar vein. I'm going to try to convince you all that I can study the first proteins and how they might have functioned. So before I get to that, I would like to acknowledge Kunal Lee and Matilda Newton, who came out of my lab. They did a lot of the work that this is all based off of. So to start off, our lab is interested, like I said, origin of the first functional proteins. So it's pretty well established by chemistry that you can get abiotic synthesis of amino acids, right? So miliary experiment, we find them on meteorites, this sort of subset of the 20 amino acids we use today, around half, 10 to 12. And then at some point, these polymerize and you have polypeptides, but they're not really functional. And then something happens, and now we have proteins, and then billions of years of evolution, and we have modern 20 amino acid based protein. What I'm focused on is this period of time, where we go from random chains of polypeptides to functional proteins that are conferring some sort of advantage, right? But when we look at these, the key thing is they don't have all 20 amino acids. They have some sort of subset of these primordial amino acids. So what are primordial amino acids? Well, we're focusing on the 20 amino acids that biology uses today, no xeno amino acids, unfortunately, but we used a consensus of the addition of different amino acids using more than 60 different theories from many different fields, mathematics, chemistry, biology, biochemistry. And then we arbitrarily create different alphabets sort of to create time shots, like snapshots of this timeline. So 20, 16, 9, and 5. And with this, we think we can answer a few big questions. So can you get biological function from primordial alphabets? What is the minimum alphabet needed for these proteins to function? And if they do function, how are they similar to modern proteins? So to investigate these questions, we created, with these 4 alphabets, large libraries of completely random 80 amino acid long proteins, so 10 trillion random proteins in each different library. And we screened for a really simple function, ATP binding. It's pretty ubiquitous throughout all life. And we kind of just saw what we could see. And believe it or not, every library actually yielded functional proteins, which we were really surprised about. We kind of aimed for the 20 amino acid alphabet library to be a sort of positive control, right? Because it's modern, we know those work. And we sort of aimed for the 5 to be a negative control, because we really did not expect those to bind ATP. But they all do, and actually the 5 performs just as well as the 20. So now we know reduced amino acid proteins, they function, but we don't know how. Especially these really reduced ones. They're lacking a lot of key residues that extant ATP binders use. So that's what I've been working on in the last year, is trying to figure out how these work. And so I've been trying to characterize them structurally through X-ray crystallography. So if you don't know about X-ray crystallography, it's just you crystallize a protein, you collect diffraction data, and you can extrapolate that to get electron density and then model that into a protein structure. So with this, we really want to look at, from some of these best binders that we found, what is the binding mechanism? how similar, how different is it to modern proteins? Because if it's similar, that's really exciting. Maybe this was sort of always bound to happen. And if it's different, maybe that means it's sort of frozen accident, and we can see, sort of sample from a completely different sequence space. And also, how dependent on alphabet complexity is protein functionality, which kind of goes back to the last talk. So with that, I'd just like to thank everyone in my lab and our collaborators from the University of Minnesota, but also from UCLA, specifically Irene Chin and Celia Blanco. So if you have any questions, see me at poster 16.
Our next speaker is Ridi Gondalikar from the Earth Life Science Institute.
Hello everyone, I'm Ridi. I'm a graduate student at the Earth Life Science Institute. In our study, we have tried to understand scaling of protein function across the tree of life and the mechanisms that might have led to the species diversity on the tree of life right here. So we study scaling using power laws, and power laws are found everywhere. If we consider the number of web hits on a web page in a given period of time, or the earthquake magnitude in an area over a given period of time, many natural and man-made processes follow power laws. So how can we how can this help us understand some concepts in biology? For that, let's consider a Lego set and consider the unique pieces in relation to the total pieces in the Lego set. When we plot this on a log-log scale, we see the larger Lego sets use more unique piece types, but they progressively go on using lesser additional piece types. So they're becoming more efficient, which means the larger sets are using the same pieces the smaller sets are using, but in more efficient and more complex ways. So what we're observing in these plots is a scaling relationship. And when we observe a scaling relationship, we could say that maybe there's a set of rules that's sort of governing the way something is working. So here's a power law equation, one quantity varying as a power law of the other. And when we plot this on a log-log scale, we get a straight line with the slope alpha. So previous studies have shown that genes in a specific functional category scale as a power law of the total number of genes in a genome. So for example, transcription regulation is almost quadratically scaling, which means if the genome doubles in size, the genes in this specific category are going to quadruple. So we have tried to include an expanded taxonomy in our study, and for that we use the EGNOG database. So after power law fitting, we saw different trends in our data for the smaller and the larger genomes. So we carried out piecewise regression to give justice to the different patterns in scaling observed in the plots. So for example, we wanted to capture the slope variability for the smaller and the larger genome sizes. So for example, in category M, which is cell wall and cell membrane proteins, so the x-axis has the total protein annotations, and the y-axis has the category annotations for that specific category. So for the smaller genomes, we can see the proteins are scaling fast, which means as the genome size is increasing, they are incorporating more and more proteins faster than the larger genomes, because after a statistically detected breakpoint, the scaling slows down. And we can see a similar trend in category T. So we saw this trend in most of the breakpoints that were supported in bacteria. Interestingly, we observed an opposite trend in archaea. The scaling is slow in the start, but fastens up after the statistically detected breakpoint. So a lot of categories were common between archaea and bacteria, but there were some categories that were exclusively present either in archaea or in bacteria. And it's also interesting to observe these differences in scaling pattern before and after the breakpoint in archaea and bacteria. So we thought maybe these differences in scaling patterns were caused by phyla-specific scaling. So we broke these domains down into specific phyla and found great variation in all the phyla for all the categories. So for example, we have category H here, which is coenzyme transport proteins, which also happens to be the most variable across all the phyla. So we thought maybe this phyla-specific scaling is causing the positioning of the breakpoints that we observed previously. So I placed these breakpoints on the total protein annotations to see if there's any specific pattern. But we can see these individual phyla are spanning the breakpoints, and there is no specific preference for the phylas to be present on either sides of the breakpoints. So maybe taxonomy is not causing the positioning of these breakpoints, and maybe there are some other factors like physiological or environmental factors that are causing these breakpoints. So we were also interested in these groups, CPR and DPAN. So these groups have extremely small genomes, and they lack major metabolic pathways. So we compared them with eukaryotes, unicellular eukaryotes, and Asgard archaea. So for some categories, we can see the scaling is very similar for category O, but for some categories, the scaling is very different, like in category C, which goes on to show there are different ways in which an organism can adapt while growing in their genome sizes. I've just discussed a few key results in my talk, so if you want to discuss, I would be interested. Please come by and stop at panel 2 for the poster. Thank you.
Our next speaker is George Shibley from the Montana State University.
Okay, so we're going to shift gears a little bit here. And so my project largely focuses on the evolution of multicellularity in bacteria specifically. So titled here, Cellular Differentiation Within an Obligate Multicellular Bacteria. So where I like to start is just generally when we think about the evolution of life on Earth, we can kind of think of it as an increase in complexity as life continued to evolve. And this is, yes, an oversimplicity, but thinking of it as molecules coming together to form a first cell and eventually getting a multicellular organism, and then something that looks like the life that we have on Earth today. If we look at our classic tree of life, where we have different domains of the bacteria, the archaea, and the eukaryotes, generally we tend to think of all the multicellular life forms belonging to the eukarya, and we think of the bacteria and archaea as these simple single-celled organisms. If we look at the tree of life as we know it today, multicellularity actually it can be plotted across the entire tree of life. And so what I'm showing here is the aggregative evolution of multicellularity, or a clonal evolution of multicellularity. And we see it present in the bacteria, the archaea, and the eukarya, of course. And so just focusing in on some bacteria that are capable of this, these are just some micrographs showing different cyanobacteria that are in those chains up there in the upper left, and then myxobacteria that sporulate, an archaea called Methanosarcina. And another example is this bacteria called cable bacteria that form these long, centimeter-long filaments and can actually move electrons along that. So the organism that I study is called multicellular magnetotactic bacteria, or MMB for short, because I just don't want to say that the whole talk. And so this is what they look like, where we have a TEM image showing where these cells are grouped together in this kind of soccer ball shape, or football shape. And then here in the lower half, just an SEM showing you what that structure looks like. So I built a cartoon kind of showing what this ultrastructure looks like. And one of the things I want to point out that pertains to the name is this magnetotaxis. And so they make these organelles inside the cells called magnetosomes, where they synthesize a ferromagnetic mineral that allows them to sense Earth's geomagnetic poles. And then they can taxi in the water column or the sediment column. They also have these carbon or energy storage granules inside them, this acellular center. So it's just one layer of cells surrounding this acellular center. And then these actin-like filaments that seem to have something to do with their structure and morphology. I've referred to them as obligate multicellular organisms, and this is rare in bacteria. In fact, the only example where the hypothesized life cycle is that they grow in size and then the entire consortia divides. And so we don't typically see this in bacteria, where typically we see this facultative multicellularity. So they can be multicellular, but they can also exist as a single cell. And that's true for all the examples I showed you with the tree of life. So here's just an image of one of the MMB from my sample site and kind of what we think it looks like MMB dividing into possibly 3. I also mentioned, moment of truth if the video works, that they're magnetotactic. Oh, let me see if I can get this to work. No, now I'm just hitting buttons. Okay, cool. So what we have here is a tube with the MMB in the bottom of it, and then a magnetic stir bar next to it. And if you just watch it, you can see them swim up out of the bottom of that tube, and they taxi towards the stir bar. These ones are going towards the magnetic north. Here on the right is a hanging water droplet, just on a cover slip, and there's a magnet to the right. And then what I've done is just move that magnet, turn it around, and they start taxiing in the opposite direction. So that's just showing their magnetotaxis. It helps when we think of multicellularity to have some criteria for what that is. And so just a list of these would be built from several cells of the same species, specific shape and organization, and synchronized growth, so that exclude things like cancer. No competition between the cells, coordinated behavior in response to external or internal stimuli, existence of cell-to-cell signaling, and then this last one that's kind of like the catch-all, metabolic differentiation or a division of labor. So with my organism, they fit these first four, and the last two are what we're kind of looking into. One of the things we found with our project was that these MMB are not actually clonal, and how we did this was we sorted whole consortia, did multiple displacement amplification, so we amplified their entire genome and sequenced that, and we were able to map reads to the longest read, the best assembly there, and look at these single nucleotide polymorphism differences. And what we found was that they have, compared to controls, which is a Pseudomonas control, as well as other environmental cosorts, they have this much higher single nucleotide polymorphism rate than what we see with other organisms, which was kind of a surprise, and we're still scratching our heads about this. And that equates to 20 to 100 nucleotide polymorphisms in the genome, and their genomes are about 8 megabases, which is about double that of E. coli. Another thing that we looked into was this metabolic differentiation, or this division of labor. And this was done by doing stable isotope incubations, where we incubated them in the presence of these 13C or deuterium oxide chemicals. And then we use nano secondary ion mass spectroscopy to look at those and map that back, where we found in the lower left, where it's the 13C image, differences in the use of that substrate, indicating this division of labor that we suspected. So with that, just to kind of give conclusions and takeaways, the MMB are the only known obligate multicellular bacteria that we know of yet. They are not clonal and do not conform to these canonical ideas of what multicellularity is, where we typically have this clonal development from where one cell divides into daughter cells that stick together, or this aggregative multicellularity, and they seem to fall somewhere in the middle there. They also engage in a division of labor, as we were looking at with the metabolic differentiation. And then, of course, just to thank the lab and all the collaborators that help us with that. Thank you.
Our next speaker is Claire Elbun from the Georgia Institute of Technology.
All right. Hi, everyone. I'm Claire, and so I'm going to talk a little bit about respiration, oxygen sensing, and this idea that there might be a really close relative to the ancestor of protomitochondria in modern oxygen minimum zones. So I think a lot of people here are familiar with eukaryogenesis. It's this idea that an archaea and a bacteria a very long time ago decided to be best friends forever, and now we're here. So there's lots of different hypotheses over how this might have happened. And kind of the main point I want to make here is that, one, there's always a bacterial host, and that's generally thought of as an alpha proteobacteria. And there's also a lot of, or has been a lot of talk historically about, is this obligately aerobic? Does it have to have oxygen? Is it facultatively aerobic, where it could use oxygen or not? Or is it kind of an anaerobic partnership that turned into aerobic partnership? So a lot of more quantitative recent research, so kind of this triangulation method, suggests that this new clade of marine alpha proteobacteria from marine oxyclines, and these are really related to a group called Iodidamonas, this is an iodide-using bacteria, might be the closest living relative to protomitochondria. And so like I said, alpha proteobacteria for a while have been thought of as that sort of bacterial component of eukaryogenesis. And what I'm going to showing you really quick right here is that these are some anaerobic traits that are thought of really essential when you're looking at the criteria for this ancestor. And so most of these are alpha proteobacteria. And when we're looking at the heat map, what we want to focus on is this green highlighted here. That's this new clade. So they hit all of these criteria for anaerobic traits when we're thinking about this protomitochondria. And then when we're thinking more about the aerobic side of this, these facultative aerobics, we want to think about the terminal electron chain. So what's interesting is that this is comprised of 4 complexes, and these complexes actually have this really phenomenal history of the evolution of respiration inside of it. So from complex 1 to complex 4, we can kind of track as the Earth went from anoxic to the Great Oxidation Event to kind of what we experience now. And so I'm really looking more at complex 4 because within it, this is that last step, that important reduction step. So if we're thinking about protomitochondria, they would need to have this complex 4. So that can be divided up even more into this A to C family. We have an A type, lots of oxygen. We don't have to bind it super well. It's there. But this C family is thought of as the oldest terminal or complex 4 of the terminal electron chain, and that's because it's capable of binding oxygen at very, very low levels. So for this new clade, I had 2 main questions looking at sort of data mining, which is what kind of genes do they have for respiration? Is it entirely aerobic or is there anaerobic as well? Are they able to sense oxygen, and are they moving along gradients in response to that? And then on top of that, are these genes actually being transcribed? So I did a lot of data mining in the with this nice big data set from the Saanich Inlet. That's in British Columbia. It's a seasonally hypoxic area, but it's an oxygen minimum zone. And that just means that very quickly we'll lose oxygen very high up in the water column. And you can see how like seasonally variable it is, but overall there's a pretty dramatic drop in oxygen. And so the 2 really interesting points I saw was, 1, there are C-type aerobic oxidases associated with this new clade alpha proteobacteria, and its transcription, or the amount that it was being used potentially, increased into the anoxic depths. And that there are 2 chemotaxis proteins, so that's an aerotaxis protein. Maybe, maybe not, it might be responding to something else. They were both being transcribed really highly into these anoxic depths. So my main takeaway is we should look at OMZs more if we're thinking about protomitochondria, because there are some really interesting alphas there that only exist right now as metagenomic assembled genomes. And it would be really interesting to really look at the full genomic capabilities of them. So I'd like to thank the Glass Lab at Georgia Tech, my funding, the NSF grad, and then the Oceans Across Space and Time team.
Our next speaker is Marissa Mayer from NASA Ames.
Hi there. Okay. Hi, I'm Marissa. I'm a postdoc currently at NASA Ames Research Center, and I work on microbial sources of hopanoid lipid biomarkers in different hot spring environments. Today I'm going to be excitedly sharing with you some new potential microbial sources of 2 and 3 methylhopanoids that we were able to find in some of our anoxic, acidic, and sulfitic hydrothermal springs. I want to acknowledge my funding sources, which is ORAU NPP program, National Park Service, NASA, and finally Stanford University, because some of this research was done back in my PhD as well. So yeah, you might be wondering, if you are an astronomer or anyone but a lipid chemist, what are hopanoids and what do they teach us in modern environments? Why should we care? So hopanoids are cyclic triterpenoid membrane lipids, so they're complex. They're a little bit different from some of the phospholipid membranes we've been seeing today. Hopanoids are some of the most abundant bacterial lipid biomarkers on Earth, so they're very worthy of studying. They are important geologically. They can withstand the lithification process and all of the diagenetic forces that happen when you are becoming a fossil. And so they're actually one of the few really strong sources through deep time of microbial inputs into the rock record. And they can be detected billions of years after deposition. Finally, in modern acidic springs, we observe methylated hopanoids. In this work, we, in modern acidic springs, observed methylated hopanoids. And we found these in an anoxic environment where no cyanobacteria and no methanotrophs were present. These are kind of the typified known producers of these kinds of biomarkers, so it was exciting to find them at all. And yeah, the work that once we found those methylated hopanoids and the follow-up work was able to lead to discovery of some new potential source organisms for these lipid biomarkers, which if you are a biomarker nerd is always exciting and it's ever expanding. So yeah, this kind of new evidence of source organisms is important because these, kind of anoxic environments and knowing about them and the microbes that live in them today can have implications for us to reinterpret the sedimentary rock record on Earth, but also potentially on Mars or on other planets where we have a sedimentary rock record. So my samples were taken in 2 sites. One is in Yellowstone National Park and one is in Lassen Volcanic National Park. In both springs, you can see the mat is kind of similar. There's a purple layer, there's like a acidic algae layer, and then there's this white flocculent elemental sulfur on top. That's from the actual hydrothermal system itself. So what I did is I go into these springs and I take samples and I go back to my lab and I extract for both DNA and for lipids. And then from there I analyze them. So I found a bunch of different hopanoids. So I did a LCMS analysis on them, and so I was able to actually look at the functionalized extended hopanoids. And so, as you can see, there's a lot of colors on that graph. Every color is a different type of hopanoid that I was able to find in my springs. The green overlay on some of them and the pink overlays on some of them indicate a 2-methyl for green or a 3-methyl for pink. So we found a huge diversity of methylated hopanoid compounds. Over to the far right, you can see my controls. We had pretty low diversity of methylated compounds, so it was really interesting in the environmental samples to see so many different kinds, which is what sent us on our DNA work. So the first thing that we did was do 16S amplicon analysis of our extracted DNA. In that, we didn't see any obvious sources or classical sources of those methylated hopanoids, which was pretty exciting. So that then led us to go back and then do some metagenomics. And then when we were able to analyze that metagenomic data, we saw many potential sources of novel, 2-methyl hopanoid producers, mostly anaerobic water and soil-dwelling microbes, but way more sources than are currently understood. And this is just from looking in 2 hot springs. So a case for doing field work to expand our knowledge in the lab. We also saw a ton of potential novel sources of 3-methyl hopanoids. These were mostly anaerobic and acidic environment dwellers that we found them tagging against. And yeah, it was pretty exciting. So if you want to know more about either lipids or DNA analyses or microbial ecology of these acidic springs, please come check out my poster later and find me. I'll be at Board 22 at this next session. And also because I am in a room full of early career researchers, I coordinate a division seminar at NASA Ames, and we would love to hear you guys give talks. So if you would like to give a talk at NASA Ames at any point, please come find me at my poster, and I'd love to write your information down, and we would love to have you give a talk. So yeah, we're friendly.
Our next speaker is Colin Robinson from Brigham Young University.
Hi, everyone. So I'll be talking about Martian metabolism and biotic perchlorate reduction in Mars analog environments on Earth. So since the discovery of, let's see how I can get this to go. Since the discovery of perchlorate on Mars by the Phoenix lander in 2008, it's been a compound of interest on the red planet. This is due to its high oxidative potential on par with oxygen, which means that it's a potential energy source for life on the red planet. On Mars, it's ubiquitous in Martian regolith, reaching concentrations of 0.5 weight percentage. On Earth, there are microbes that reduce perchlorate in place of oxygen in metabolism, using it to drive the electron transport chain. And perchlorate accumulates in Mars analog systems on Earth, such as the Atacama Desert, where it reaches concentrations up to 0.1 weight percent, as well as the Antarctic dry valleys, where it reaches concentrations up to 0.003 weight percent. These are Mars analog systems that are incredibly dry and accumulate perchlorate via atmospheric deposition over long time scales. And they're likely, looking at these systems, it seems reasonable that perchlorate-reducing microbes might evolve and persist in these types of environments. However, the model organisms that we use to study biotic perchlorate reduction don't come from Mars analog environments. They come from the Potomac River in Washington, D.C., where there's very little to no natural perchlorate that accumulates. All the perchlorate that we do find here is waste from munitions factories that gets dumped into the river. So how did perchlorate-reducing microbes arrive in this type of environment if they likely didn't evolve here due to the lack of perchlorate? Well, we know that the perchlorate reduction genomic island is widely spread via horizontal gene transfer. We know this because when we look at perchlorate-reducing organisms, can you see my mouse? Yeah, there we go. The elements required for biotic perchlorate reduction, the genes that are required for perchlorate reduction, share a lot of homology between the genes, but where the genes are situated in the genome is very diverse, depending on the organism that you're looking at. So it's a very chimeric metabolism. So if we look here at the bright green, that's chlorite dismutase, and this long dark green, that's perchlorate reductase. They're always kind of in different configurations in relation to each other in the genomic island. And that's because these individual elements are getting passed individually and independently of one another via horizontal gene transfer. So you only get perchlorate-reducing microbes when these elements come together in the same organism. So my research question was, did canonical biotic perchlorate reduction arise in these Mars analog environments like the Atacama Desert or Antarctic dry valleys, and then disperse around the world via acellular and microbial dispersal? So in order to test that, I selected metagenomes from sites around the world. A metagenome, for those of you who don't know, is basically all the DNA in a sample. It comes in little 20 base pair chunks, and you have to piece them together like a puzzle, and then pick out the puzzles that you're interested in, that maybe make a picture you're interested in, and then analyze those independent of the rest of the metagenome. So basically what I did is I selected metagenomes from sites around the world. I analyzed the presence of perchlorate reduction genes, so basically built the little puzzles and then pulled out the ones that made perchlorate reducing genes. And then I calculated the phylogenetic diversity of those genes at each site. This is a study that has really up until this point been done only in humans, which is this a type of analysis where we look at diversity of a species or a gene to determine the likely places where it's persisted the longest. That's how we know that humans come out of Africa, is because most of our phylogenetic diversity can be accounted for in Africa. So when I did this, so this is for chlorite dismutase, a gene that's involved in perchlorate reduction. I found the highest diversity to be in the Antarctic dry valleys. So what we're seeing here is these bars are are a scale that's Faith's phylogenetic diversity. 0 is basically 0 diversity, and then up to 50 would be the maximum amount of diversity in these genes that we see. And each of these blue dots is a separate metagenome. As you can see, there's a little bit of sampling bias because in the Antarctic dry valleys or Atacama Desert, we have a lot more sampling. There's a lot more research that happens in these sites. We've got a lot more metagenomes that I can analyze. Whereas if I look at other Mars analog sites like the Namib Desert, there are quite a few less, or in South Australian Desert, much less access to that kind of data. When I looked at perchlorate reductase, another gene involved in biotic perchlorate reduction, we also see that it reaches highest levels in the Antarctic dry valleys. So that basically indicates that these genes have persisted the longest in these Mars analog ecosystems, supporting the claim that these may have evolved and persisted in those Mars analog systems. So some conclusions: perchlorate reduction genes are present and diverse in Mars analog systems. That's really awesome, right? Before, we didn't even know if they could persist in Mars analog systems, but it looks like they both evolve and persist there for long time scales. This metabolism may have evolved in these environments and then prior to dispersing around the world via acellular and microbial dispersal. And studying perchlorate-reducing organisms in Mars analog environments can give us further insight into how life may persist on the red planet. When we're studying life forms that do metabolisms like what we expect to find on Mars, we shouldn't be looking at organisms that come out of the Potomac River. We should be looking at organisms in these Mars analog systems. So I'd like to hear some people I'd like to thank. So my funding, which is the NSF grant for long-term ecological research at McMurdo Station, my department at Brigham Young University, as well as the help and advice of Dr. John Coates at UC Berkeley, Dr. Kendall Lynch at the Lunar and Planetary Institute, and Dr. Jennifer Glass at Georgia Tech. So thank you.
And our last speaker is Silath Pandey from GPL.
Hello everyone, I'm Siddharth from Jet Propulsion Laboratory, and I'm here to talk about the work that I started before joining JPL in India. And for those of you in the audience who are currently involved or plan to do work in different parts of the world where astrobiology has huge potential and significance, hopefully with some of my experience, you'll be able to take things forward. So first of all, a little bit about myself. So I'm originally from India, and before joining JPL, I have an engineering background, and my current work is actually on Enceladus, on sample collection technologies. But unfortunately, today I'm not going to be talking about that. This is primarily going to be focused on that. But just giving a heads up, people can find me after the talk, and maybe we can chat. But here is why we focused on India. Well, first and foremost, as a country, India has one of the largest student populations, as some of you might be aware. Every year, we've got several thousands of students graduating in science and engineering, and on a daily basis, we get inquiries about how do you study and how do you work in astrobiology. So there's a huge supply and demand problem, and there's little to no formal education or career prospects in the country. So that was the first and foremost no-brainer reason. Secondly, I would like to really promote the fact that we have an amazing diversity of high-value science analogs. And these are places where some of you might actually find some of your work to be super relevant. So with this in mind, these sites are not even on the atlas of astrobiology sites that the community normally works in. And lastly, our National Space Agency, Indian Space Research Organization, is finally reaching a point to involve astrobiology as an important science priority for its future missions. It had the Mars Orbiter mission in 2014. Currently, it's under works to work on a surface mission that would work on the progress and the success of the lunar mission that will be launching in a month from now. And finally, there is the Gaganyaan program, which is the human space program, which will have astronauts working in low Earth orbit. So with that as the basic motivation of why we decided to do this 7 years ago, let me talk quickly about the background in terms of what astrobiology was happening before we kind of stepped in. So the earliest what we could find is between 1997 to 2005, ISRO was involved in scientific balloon experiments where they were basically working on collecting and understanding microbes in the stratosphere. Cut to 2015, where as a grad student, I got an opportunity to go on an astrobiology expedition to New Zealand. And along with a few other Indian and Indian origin researchers, we decided to bring the program to India. And that's what we did. As part of the NASA Spaceward Bound India expedition, that was the first time that a dedicated astrobiology expedition was taking place, gathering and bringing together researchers in the country. And that's where ball just really started rolling. The momentum started building up. And let me tell you something, that working and socializing with people in uncomfortable, dry, desert environments is way better than inside an air-conditioned classroom with experience. So some of the things that led from that was the establishment of a Center of Excellence in Astrobiology at Amity University, Mumbai, which was the first dedicated center that I was part of leading. And also as part of Blue Marble Space, the organization that I had been affiliated with, the Astrobiology India group has been active since then, working to promote coordination activities between domestic researchers in the country. So we've been having national level meetings and discussions. And what we want to do from here is move forward towards setting up a national astrobiology program closely in support with the Indian government. I don't have enough time, but I'll try to do justice to this slide. Our efforts at the center focused on 3 main verticals. The microgravity research, we built plant-based plant growth observation experiments for ISRO. We worked on characterizing some of the sites in Ladakh, which is an early Mars analog, and hardware testing for rovers. And in collaboration with UC Berkeley, the Berkeley SETI Research Center, we had 4 research fellows who were working to use radio telescope data from GMRT, which is an array facility there, to set up a SETI pipeline. And our research work has culminated into the university finally agreeing to have a master's program that is dedicated for astrobiology and science students. So hopefully this year onwards, we'll have students in India who will have that opportunity to study there. Blue Marble Space has been a great source of information and help and network. So those of you who are not affiliated, I would say strongly recommended. Do consider it. Quickly talking about some of the unique aspects of astrobiology in India. I've just highlighted some of the main sites here in Ladakh, up north, which is a high altitude cold dry desert environment. Kutch, which is one of the largest salt flats in the world. The only accessible basalt impact crater in Lonar, the basalt traps and limestone caves, and there are several others. And because we don't have a National Astrobiology Institute yet, there have been institutions that have been mushrooming over the last few decades. And I've been very successful and fortunate to be part of several senior Earth scientists to work towards setting this up. And it's only because of these people who you're seeing on the screen over here that we're able to do this. And we've been working through the pandemic to make this happen. There've been a working group, several working groups that have been put in place. We've been closely following work done in Europe and in the United States through the NASA Astrobiology Institute to help form these working groups. And these are just some names and some areas where people have been working. And too much information, bad slide, I know, but I just wanted to tell you that there are these sites, and we have been trying to map out the science objectives, the different technologies, what worlds are they analog to, and what kind of missions would they be able to support into the future. Some other ways in which we have been contributing, we published our first advocacy paper in New Space Journal talking about the importance of astrobiology for India. And internally, we have been contributing to the science roadmap for ISRO as well. Now, 3 main wins for us. One is the fact that we've got several students who have gone through our system, and these are the people who in the future will be taking the light forward. This year, ISRO published its space policy document, and we were very happy to see that they have acknowledged the importance of astrobiology because they've mentioned that they will be looking into extraterrestrial habitability. So for us as a country, this is a huge win that finally ISRO has acknowledged us. And the fact that there is now comprehensive, I would say not enough, but still comprehensive diversification of funding for scientists in the country to do this, but definitely more work needs to be done and more funding needs to be put together. So my last slide, to summarize some of these things, if you're in your region or country interested in setting things up, the first and foremost step is to bring early and mid-career people together and encourage conversations to take place at a domestic level, at the same time leveraging international work. And like I said, field workshops are amazing for team building. Finding an important university or a host institution is critical because if you're doing events, logistically speaking, it's important to find an organization that will support you. And beware of non-scientific and exploitative groups in the region. Finally, convene workshops, identify research interests, try to tie all of them together into overarching themes, and help the group formalize themselves in the form of a virtual network, a nonprofit, or a society. And finally, encourage the group members to work closely with their government for their astrobiology goals. So that's my time, and hope you can find me, and those of you who are interested can talk more on this. Thank you.
Okay, so that concludes the first short talk session. So we're going to start our poster session now. Be sure to find all the speakers and others, to talk to them at their posters. Just a couple housekeeping things. Our friend's defense party is happening in Surfside right now. So we've moved a lot of snacks and coffee and all that just outside in the courtyard. So if you have a couple things that you really need to grab from Surfside, just do it quickly and quietly. I think they're showing up at 1:30. But for the next couple hours, really, we're just going to be in the forum. We can go outside on the green space, but let's just make sure that they can celebrate in Surfside. And then one thing with trash. We need to, I guess we can't put too much trash in there because I guess UCSD catering usually takes care of that. So we've been told to we'll put out some other garbage cans, but just refrain from using those as much as possible. Yeah, that's all. Thanks, everyone. All right, everybody, we're going to start our Salty Stuff talk session, the last one of the day. And our first speaker will be Emily Paris from Stanford University.
Hi, everyone. I'm Emily, and I'm a PhD candidate in the DECUS lab at Stanford University, and I study the limits of life in hypersaline environments. So as we all probably know, brines are one of the targets for a life detection mission to other ocean worlds, including Europa and Enceladus, and even past ocean worlds like Mars. And so by understanding the limits of salty life on Earth, we can understand how and where to look for life on these other places. One of the ways we can assess a brine environment is through this measurement called water activity. And water activity depends on vapor pressure, but you can think of it as how many water molecules are available to a cell in solution. So on the left is an image of what pure water would be. This is where all the water molecules are free to become vapor, move about the cabin. That would be a water activity of 1. Now, if there's ions in solution, water would be attracted to those molecules and a water activity would decrease. So the lower the water activity, the more uninhabitable an environment is. And since water is required for all life as we know it, water activity can be used to assess habitability. So some common items and their water activities are listed on the left, including seawater, which has a pretty high water activity, and the salinity of seawater is about 3.5%. And then honey has a water activity of 0.6, and some of Mars' special regions or areas where we might detect life on Mars are no lower than 0.5. And there's been a lot of really great pure culture work on the limits of life and cell division at different water activities. And to sum it up, the predicted or theoretical water activity limit for all life on Earth, including eukaryotes and prokaryotes, is about 0.6. And this is based on pure cultures, so like our speaker last night mentioned, not all the things in the world are in pure culture yet, so that leaves out about 90% or more of the biodiversity on Earth, and our ability to assess what the limit of life for each of those strains of microbes are. So brine environments are really widespread on Earth. They're everywhere. And a lot of great work has gone into assessing metagenomics, like the taxonomic diversity, the potential metabolic potential of microbes in these environments, metatranscriptomes, and even some bulk activity measurements. But metabolic activity is rarely measured at the single cell level. And this can be really important because when a cell is stressed out, they might have really low levels of activity that are not detected with bulk techniques. So today I'll be talking a little bit about my work in solar salterns and then transition into my current work in Western Australia acidic brines. So, like I mentioned, when life is reaching its limit, metabolic activity can be really low. And so single cell analysis can help us detect this low level of activity by lowering detection limits. So a cell needs to acquire 50% new biomass to be able to divide into 2 new daughter cells. However, with nanosims, for example, we only need to detect about 5% maximum. We can even get as low as less than 1% of new biomass to detect active life in a sample. It also provides quantitative results, so we can do a lot with this data on a quantitative level, and it also reveals trends that are often obscured by bulk analysis. And when I'm talking about metabolic activity in this talk, I'm talking specifically about anabolic activity. So there's catabolic activity that produces energy for a cell, and then there's anabolic activity where a cell can take substrates from the environment, like amino acids and glucose, and take that carbon and nitrogen, and then produce new cell biomass, which includes proteins, DNA, and cell walls, and that sort of thing. So first, the work that I've done at this solar saltern, it's called South Bay Saltworks. It's here in San Diego. And basically, they bring seawater in from San Diego Bay, and it goes through a series of evapoconcentration ponds. And as the water evaporates out of the seawater, different salts are left behind, producing a series of ponds that range in water activity from 0.98, that of seawater, to that well below the known limit of life at 0.4. And so we measured anabolic activity of over 6,000 individual cells from a series of 5 of these brines, with 5 different isotopic substrates from amino acids, ammonium and bicarbonate, glucose and nitrate, and that sort of thing. We measured their anabolic activity with nanosims. So on the left, this is what this looks like. Cells that are circled in red are considered enriched, and this is for carbon from amino acids. And then the cells that are circled in white would be considered less active or less enriched in this sample. And when you plot this with water activity, you see that microbial activity decreases exponentially with water activity. However, there's certain areas, let's see, can we see this? Yeah. Certain subsets of cells are more active than most of the cells in seawater at a higher water activity, with even just low decreases in water activity. We also detected cell biomass through DAPI and nanosims in this low water activity magnesium chloride brine. However, we didn't detect any activity here. And the incubation time was 2 days long, so there's several reasons for this. We were also able to use this data to estimate a new predicted lower limit of life at 0.54, so that's a little biologically significantly lower than the 0.6 previous limit predicted. And this data, I'd love to talk more about it, but it's been submitted, and I just got word that it's now in review as of this morning, so that's really exciting. And if you'd like to hear more about that, please talk to me later. So now to kind of get into the topic of this talk, which is talking about the limits of life in acidic brine environments. There's a lot of things that happen in brines. So you can have salinity as a limiting factor, you can have high metal concentrations, you can have high ionic strength and chaotropicity. But one thing that really stood out to me about this sample set was that most of the brines that had high levels of activity actually had a pH about 7 or 8, so like relatively neutral to slightly basic pH. However, this magnesium chloride brine, which had no detectable activity, had a pH of about 5.4. And so my question was, how does pH affect microbial activity in brine environments? And is this an important factor in the limits of life? So last summer, our team went to Western Australia to sample a bunch of acidic brines. And these brines are considered Mars analogs because of the geochemistry and weathering systems that created them over millions of years. Just in short, oxidation of sulfides acidified the water, and then evaporation concentrated ions and other salts over time, creating these really acidic briny environments. There's hundreds of these lakes, and we were really lucky to sample a really broad spectrum of them. So on the x-axis is water activity, and on the y-axis is pH. And I was really thrilled to get a nice group of lakes to sample for activity analysis, especially these ones that are boxed. These were at a water activity of 0.7, and they range in pH. So it's a really good opportunity to kind of look at how pH affects metabolism at low water activity. So with this study, I analyzed metabolic activity, specifically anabolic activity, by incubating these brines with an analog amino acid called HPG. Cells that are active in the sample will take up HPG and incorporate them into their new proteins that are produced. And then the HPG has a alkyne group on the end that later on in lab you can perform click chemistry on and click a fluorophore to it. So all the cells that are active in the sample turn out green under the microscope or in flow cytometry, and then that can be counterstained or compared to cells that are stained blue via DNA stain of DAPI. And so with this data, you can calculate the percent of active cells across these lakes. So this is just an example of what this data set looks like. These are just 4 of the 13 or 14 lakes that we were able to do this with. On the left, the lakes are ordered in decreasing pH. And I thought it was also kind of interesting, the color changes, so you can admire those. I don't know why they do that, but that's the way it is. And then in the middle are the DAPI stained cells under the microscope, and then on the right are the active cells that are tagged with the green fluorophore. And you can also notice like the different cell morphologies and abundances that change with pH, which is really fascinating. And also that pH didn't affect the assay itself. We do see some cells that are active at these low pHs, and sometimes you have brines where there's less activity happening there. So when you calculate the percent of active cells and you put this all in a plot, I'll just orient you. So on the x-axis here, we have water activity decreasing, and on the y-axis is pH decreasing in this direction. So you would expect the most extreme acidic brines to be kind of in this region. This dotted line is that newly predicted water activity limit of life that I talked about earlier. And then this dotted line represents the percent of active cells dropping below 50% at this pH. So this color bar, each of the dots is one lake, and the color bar was colored on a green gradient if the percent of active cells is above 50%, and on a gradient towards black if it was below 50%. So you can kind of just visually see that all of the green, like high percentage active cells cluster up here, and all of the low % of active cells clustered down here. And when you look at it statistically, there is a moderately stronger correlation with pH and activity compared to water activity and activity. So many times, you can count how many times I say activity in this talk. And so some thoughts on this. A pH of 6 is really high for environmental samples. And as we've heard from a lot of people, and as we know, there's a lot of microbes that live in way more acidic environments. So what's actually happening here, I don't have an answer to that yet. And I'm hoping to figure that out with future analyses that include NanoSim's work on these lakes. And then if this is a true signal and this is what is going on in the environment, then microbial activity may be detected at lower water activities than we know of now, as long as the pH is within the 7 to 8, maybe like not so acidic range. So figuring out what that pH range is and how that interacts with water activity limits of life is something of ongoing investigation. So with that, I'd like to acknowledge my lab, the DICAS lab pictured up in the right, all of our funding sources, the Oceans Across Space and Time team, which is a NASA-funded project you'll hear about. You'll hear from a lot of us throughout this conference, and all of our collaborators in Western Australia, including Molly the dingo, who, of course, this work would not have been possible without. All right, and then I'll take any questions, and I'll leave this up so you can stare at the dots for a little bit longer.
Thank you, Emily. Any questions?
Hi, that was a really cool talk. Thanks so much. I was just wondering if you could expand a little bit on why you concentrate only on anabolic activity as opposed to catabolic activity.
Mm-hmm. Yeah, so let's see. Scientifically, the reason why I would focus on anabolic activity, I mean, both catabolic and anabolic activity are essential for life, right? Life has to make energy to assimilate substrates and grow and divide. So both processes are really important. And they are processes that I'll be combining or looking at in future field sites. With anabolic activity, the interesting thing here is that you could imagine that when a cell gets really stressed out, it would stop dividing potentially and still accumulate substrates to repair DNA, repair cell walls, do all that stuff until conditions get better. And so the idea was that, or my original hypothesis was that that anabolic activity would extend beyond the known cell division limit of life, because even as cells would be able to survive just by taking up materials. And so that taking up process is the anabolic activity part. Yeah, good question.
Sweet talk. I have a question. So you mentioned that you guys have essentially like extrapolated a new like limit for life, like in terms of water activity. It's kind of a power move. So
We're not the first ones to do this. So.
Okay, but could you explain how you extrapolated that? Because I think maybe I missed that or I didn't understand.
Yeah. Okay, we're gonna get into the weeds here, but let me go back to that slide. So this idea or this like extrapolation is based on this study here. So they did the same thing, and this is how they got this theoretical limit of life that has now been cited almost 200 times from food preservation fields to life detection fields. So hasn't been published yet. Yeah. So they used a different technique, and they based it on cell division. And so so hang on, let me answer your first question. So the way that we did this is single cell analysis allows you to separate the cells that are active from those that are inactive. And in a brine environment, this is really important because brines are really salty and cells can fall in from anywhere, bird poop, wind, whatever, and get preserved, and those can be inactive. So those can contribute to that bulk analysis and not necessarily even be important in that environment. So with single cell analysis, we took out the active cells, averaged their anabolic activity, so we're just focusing on who's contributing to metabolism in that environment, and then use detection limits and math to extrapolate and predict or propose this new predicted limit of detectable life. And so this is not the limit of life. This is what our measurements can detect. And if we found an environment at 0.54 water activity with similar conditions to this environment that we extrapolated it from, we would technically be able to detect life in that environment. And this is also a really conservative limit. So we even upped the number of standard deviations that we would normally do for our detection limit. So if we were to do our data processing in the normal way, it might even be lower. That kind of helps. Yeah. Yeah. Mm-hmm.
Hi, George Scheibel from Montana State. I'm just curious, you're looking at the active cells. Did you look at who they were at all, whether that was by FISH or sequencing?
So in this study, we did not look at cell identity, and we focused on metabolism. In Western Australia, I don't have plans to look at the, well, I don't know, I could look at the active fraction of cells from the Boncat analysis. I haven't decided if I want to go down that road yet, but in the next field site, we are planning on doing that. So yeah, not in this study.
All right, thank you very much, Emily.
Yeah, thank you.
Okay, for our next speaker, we have Zoe Havlena from the New Mexico Institute of Mining and Technology.
Yeah, New Mexico Tech for short is fine. So hi, I'm Zoe Havlena. I am a PhD candidate from New Mexico Tech, and I would like to just jump right in and introduce my field site for this project, which are two cave systems located in central Italy. So pictured here, they're very pretty. And what I can hopefully show you, I'm going to take us on a quick field trip there, is why these are potentially very compelling sites for astrobiology or for geomicrobiologists such as myself. And so these caves form through a somewhat rare mechanism, which is known as sulfuric acid speliogenesis. So this is a cave building process that's seen only in a handful of caves worldwide, and only a fraction of those can we actually access those caves and see this process actively happening today. And so that's what I'm showing you here. So these are all images from one of my field sites, which is a Frisassi cave system. And what you're looking at in this image here on the left, this is a stream. This is the groundwater level of the cave. And it's enriched in hydrogen sulfide. And this hydrogen sulfide degasses where there's voids in the host rock of the cave. And this hydrogen sulfide fuels very robust microbial communities that are thriving off of the energy in this otherwise very allogatrophic environment. And this is seen kind of conspicuously in the forms of these charismatically named snotite biofilms, which proliferate in these areas of the cave. Here's another picture of what they look like. And we also sometimes see, if you can see the yellow dots, native sulfur that can precipitate in these areas as well. And these are primarily composed of Psittithiolbacillus, which is a sulfur oxidizing bacteria. But what I'm interested in for the context of my project are all of these white, I don't know if you can see my pointer here, these white clumpy minerals here, and this is actually microcrystalline gypsum. This precipitates in part kind of as a consequence of this microbial activity because these sulfur oxidizers are producing sulfuric acid, which is obviously highly corrosive when you have carbonate host rock. And so this gypsum is precipitating as a replacement as this host rock gets corroded, and it's highly acidic, so pH is usually 0 to 1. And what happens is that this gypsum can build up in these cave systems and remain there essentially as long as you don't have any waters that come in and wash it away. And so that's what I'm showing on this image on the far right here. This is one of these massive gypsum deposits that exists in a part of the Frisassi cave that hasn't been exposed to this active sulfide degassing for maybe 100 to 200,000 years. And so we see sulfates and gypsum on other locations, notably the surface of Mars. So I'm showing you data here as it relates to the Gale crater. And there's some evidence or some thinking that this sulfates could have also precipitated under acidic conditions. So potentially these are thought of as places where we might look for evidence of life. And on the Earth's surface, the analogs that we have, there are several, and these have been well studied by other teams of astrobiologists. But what is novel, or the argument I'm making for these caves, is that this is an ecosystem that, as I just showed you, is fueled by chemolithoautotrophy. So these kind of alternative lifestyles. And so potentially studying the gypsum and its ability to preserve or not preserve biosignatures is kind of a novel aspect and gets away from surface world where we sometimes see the signatures mostly of things that are photosynthetic. And so in the context of this talk, when I'm talking about life detection or I'm talking about signatures, I'm talking about lipid biomarkers specifically. And so what I'm showing you here are some types of these. They were kind of explained earlier. These ones on the left, which are those hopanoids. So these are produced by bacteria, and these can be functionalized. And then they have this very recalcitrant backbone structure, this hopane, that is this diagenetic structure that can persist in some terrestrial environments for potentially millions of years. And then on the right, these are GDGTs from archaea, so kind of a similar lipid biomarker. And these specific structures that I'm showing you are ones that have been classified from those snodite biofilms that I showed you in the earlier image. And so knowing what we already know about these cave systems, which is that we have gypsum that is precipitating in very close association with microbial activity, that's kind of these microbes that are using an alternative lifestyle. And we know that some of these organisms, like that acidithiobacillus, are capable of producing these lipid biomarkers. And this gypsum is kicking around in this cave for a little bit of time. We sort of used what we know to form these questions about things that we don't know. We don't actually know, or going into this study, no one had ever looked at what microbes are colonizing the surface of the gypsum itself. And we don't know if those organisms produce their own lipid biomarker suite, if we'll be able to characterize that or detect it in the gypsum at all. And then if we do, does that persist in this gypsum that has been existing in this cave for a longer period of time? And so I won't go into the weeds on these methods here, but I just want to highlight that we're using kind of a few different approaches to answer some of these questions. So using some sort of classic molecular biology techniques, amplicon sequencing, and metagenomics to characterize those microbial communities, and then using some organic geochemical techniques to try to characterize those lipid biomarkers. And so some of the data that I've had is that these gypsum surfaces are in fact colonized by microbes. And so what I'm showing you here is amplicon sequencing data, so that's 16S RNA sequencing that tells us who's there. And what I want you to take away from this graph is that I'm plotting two things here. On the left, this axis in the blue dots is showing the percent of that acidithiobacillus, so that's that main sulfur oxidizing bacteria that we see in those snodite biofilms. And then on the right, I'm showing in this orange, I'm showing you the overall diversity of these samples. And so all of these dots are gypsum samples that we're taking at kind of varying distances from that actively degassing sulfitic stream. And just a reminder, so these snodite biofilms that have been previously characterized are around 70% acidithiobacillus. And so the main takeaway is that our samples of gypsum are far more diverse than that. You know, even at this kind of like best case scenario where we're the closest to the sulfitic stream, we're really not seeing anywhere near 70% acidithiobacillus. And so this is data from that other cave that I've studied, and I don't have that nice distance gradient, but again, it's plotting the same things, and I've just kind of broadly lumped these samples together in terms of like high level of sulfide, medium level, lower levels. And again, we see a similar trend. We also see low levels of acidithiobacillus, and in fact, we see a heck of a lot of archaea too. So some of these samples were almost entirely archaea. And why I talk about acidithiobacillus and I talk about the diversity of these samples is that, we know Acidithalobacillus is capable of producing lipid biomarkers, but it's one of the bacteria that can, and in fact, a lot of them can't. And so this potentially has implications when we think about, are there things that is in this gypsum that is producing lipid biomarkers or aren't there? And we can kind of get at this indirectly through metagenomics. And so again, metagenomics is looking at all of the genes that are present in a particular environmental sample. And so far, I have only reconstructed a single metagenome, and this is from a native sulfur deposit. But I did do some gene searching, and I was looking for the squalene hopane cyclase gene, which forms a critical precursor to making those hopanoid lipid biomarkers. And I did find some copies of that. But again, I also found a ton of archaea, and 2 of the high-quality bins that I was able to assemble were these 2 kind of novel archaea that have potentially an interesting symbiotic relationship. And I would love to talk about that more, but that's kind of a whole other talk. So I'm just going to leave it at that with this complicated figure here on the right. You might be guessing or kind of wondering why I've only gotten 1 metagenome so far, and really it's because the overall biomass in these gypsum samples is very low. And so what I'm showing you in this picture here, so this is microscopy, this is DAPI staining, so looking at cells. And so you see, hopefully you can see just a few blue dots, those are cells, and on the right, this is showing them sitting in a bathtub full of gypsum microcrystals. And so when you just kind of contextualize this low number of cells in the vast array of all of these minerals, there's not a whole lot going on. And in fact, some of our preliminary total organic carbon measurements are less than 1% or even a fraction of a percent. And so that all leads to kind of some complications with trying to actually extract some of these lipid biomarkers. And so this side of the project is still in active protocol development. We haven't seen anything yet, and so we've had to kind of scale up our extraction. We're hoping to extract a lot more gypsum, and I'm hoping that that's something I'll be able to talk about more in the future. But I just want to return to sort of the guiding question of this study, which is, looking at these very interesting microbial dynamics, the way that this gypsum is formed. We see biofilms existing where the gypsum is forming, but we don't know if there's biomarkers that are preserved or not. And I have to leave that question open at this time, but hopefully I can answer it in the future. So I think I will just leave off there, and I wouldn't be able to do this work without, I have a lot of very fantastic collaborators and funding sources, and I would especially like to thank all of the folks at the Astrobiology Analytical Laboratory at Goddard, and I think that I have some time for questions. So thank you.
Thank you, Zoe.
Hey, I'm Bonnie Chase. I'm from the Jet Propulsion Laboratory. I really love this question you're asking and like the way that you're trying to attack it. And I was wondering, a lot of the lipid biomarkers that you talked about looking for, they, as you said, can be preserved for a few millions of years. But what about the more stable versions, like looking for hopanes rather than hopanols? Are you also looking for those?
Yeah, we are. I didn't really explain that super well, but we are hoping to look for hopanes as well. Yeah.
And what do you think, like, the preservation pressures in this cave would be for the molecules, and how similar is that to Mars?
Yeah, I think that is kind of the key question for sure. I don't know if that's something that I want to dive into right now, but I think we should talk about it later for sure. I'm curious to hear your thoughts on it as well. Obviously, this is an oxidizing environment. It's acidic, it's wet, so it has a lot that could maybe not be preserving things, but certainly folks have done similar research and have found biomarkers in in gypsum in other environments on Earth. So it's a good question.
Any last questions for Zoe? Okay, thank you so much. Oh wait, there's Chad. Sorry.
Hey, so I have a question about the microcrystalline nature of that gypsum.
Yeah.
So what do you think that might, what's your hypothesis for structurally how that might affect preservation of biosignatures as opposed to other kinds of gypsum that might happen on the surface?
Yeah, I think that that's a really good question. I guess, off the top of my head, I would say that I don't know if we're gonna get, it like doesn't really layer, I think, necessarily in the way that we see in like evaporitic gypsum. How that may or may not, factor into biosignature preservation, I can't really speak to that, but I think it's definitely a really interesting difference that we see in this gypsum for sure. And it's like, I didn't really show any of the, like, there's that one picture that I showed of the gypsum, but it's crazy. It's like, we call it rock in name only. It's like goo. You go to sample it, and it's almost like a non-Newtonian fluid. It like just kind of like oozes, which is super wild, and it's kind of difficult to work with, to be honest. Yeah.
Thanks.
All right, thank you so much, Zoe.
Thanks.
Okay, so our next speaker is Kimberly Sinclair from the University of Washington.
Hi, I'm Kimberly Sinclair. I'm from the University of Washington, Department of Earth and Space Sciences. I'm working with David Catling and Tim Elam. I'm going to talk to you guys about this project, the mineralogy of evaporites and sediments in the alkaline phosphate-rich lakes of the Caribou Plateau, which is work I've been doing with Sebastian Haas and is funded by the Simons Foundation. Okay, so I want to give you a bit of background about the phosphorus problem in astrobiology, because I know we're all coming from very different backgrounds. There's a lot of biology talks this morning, though, so I'm sure a lot of you guys are caught up on this. But so we all know DNA on the right-hand side here has this sugar phosphate backbone, and then you have your base pairs. Or if you zoom in closer, you're seeing your phosphate group, your sugar, and your nuclear base here. And in order to get this phosphate to incorporate into the nucleotide, you need very high levels of phosphate. And this has been shown by research done by Pauner et al. In 2009. So this question we want to ask is how much phosphorus is actually available for prebiotic phosphorylation on early Earth? And even though we need these very high concentrations of phosphate, up to 1 molar in prebiotic phosphorylation experiments, we only see very low concentrations of phosphate in the environment, close to the micromolar level. And that's because in natural waters, phosphate will combine with calcium and fall out of solution as an insoluble mineral known as apatite or calcium phosphate before reaching very high concentrations. So there's a bit of a problem here, and that's what we call the phosphorus problem. We've proposed a solution to the phosphorus problem. In a paper by Toner and Catling in 2020, the hypothesis was stated that abundant carbonate leads to calcite formation, calcite being calcium carbonate, thus suppressing the calcium phosphate mineral formation and leaving phosphorus in solution to concentrate to higher levels. So in order to test this theory, we visited the most phosphorus-rich lake in the world. This plot on the right-hand side is from Toner and Catling 2020. You see very high phosphorus concentrations in these lakes on the top right side, which are good enough in Last Chance Lake. And so that's where we wanted to visit to understand why you're getting these very high concentrations of phosphorus. We can then apply our understanding of these lakes to possible analog lakes on prebiotic Earth where origin of life could have happened. So a little bit about these lakes. Last Chance and Goodenough lakes are carbonate-rich alkaline soda lakes on the Caribou Plateau in British Columbia, Canada. Left-hand side, you see the map of British Columbia here, and this green swath is the Caribou Plateau. It's right east of those coastal mountains, not too far north of Vancouver. If you were to zoom in on Google Maps, you would see the lakes here, Goodenough Lake here, and Last Chance Lake here. They both have very high phosphate concentrations, but particularly Last Chance Lake gets up to 37 millimolar of phosphorus, which is very high in comparison to that micromolar level that we're usually seeing in natural waters. And this is an image of Last Chance Lake taken in November 2021, which is what it looks like from the ground level. So you're seeing it form into these sort of brine pools. So a little bit of context from these lakes. They're 2 small, less than kilometer squared, shallow, closed basin lakes with sodium carbonate, sulfate, chlorine brines. These are typical major ions of soda lakes developed on basaltic rock. Their sulfate, potassium, phosphate, and magnesium are abundant, whereas calcium, iron, silicon, and dissolved inorganic nitrogen are present at very low concentrations. The salinity reaches up to 462 grams per liter, which is about 13 times seawater salinity, so they're very salty. And the pH ranges from 9.7 to 10.7 in both of these lakes, depending on the time of year and the location in the lakes, because they do get spring infed from surrounding rivers and such. Or sorry, from groundwater, not rivers. So we wanted to go and visit these lakes over different seasons in order to understand how the mineralogy of different precipitates and evaporites, as well as precipitates in the sediments, change. So we went in winter, summer, and fall, and you can see the corresponding pictures here. In winter 2021, you see these brine pools starting to freeze over. In summer 2022, it enters this ephemeral lake stage where it has enough water that it actually covers the entire surface. And then in autumn 2022, these brine pools are drying out into salt flats, and so there's basically no water left in this system at all. It's just salt. So we wanted to collect lots of different evaporites, which are these salts that are forming around the lake shore, as well as precipitates within the lake. These are different fun examples of evaporites we collected. So in our winter trip, you get a lot of these salts forming on rocks around the lake shores or around the brine pools. In June, this is an example of a precipitate that formed and fell to the bottom of the lake, and so you can pick it up from under the water. It's a lot of salt mixed in with mud and clay. And then in September, when it's in that salt crust phase, you can dig into the salt crust and take this out, and that's what the cross-section looks like. So you're getting these very white, opaque salts on the top, that middle layer of translucent natron salt in the middle, and then a layer of algae right above the mud. We also wanted to collect sediments from the lake bottom, and so these are pictures of us collecting different sediment samples with sediment cores. We did it both around the lake shore as well as at the lake bottom, and then the center picture you see is us taking those sediment cores and chopping off different depths in order to see how the mineralogy changes with depth in the sediments. So we did mineralogy analysis with X-ray diffraction. We used a Brooker D8 powder X-ray diffractometer with a copper anode microfocus X-ray source, which is the microfocus X-ray source is really useful when you have very small quantities of powder that you're trying to analyze. And a Pilatus 100K large area 2D detector is really helpful when you want to increase the resolution of your diffraction patterns. Just a quick note about sample preparation. We tried to isolate the mineral phases visually as much as we could from our samples in order to simplify the pattern matching. Grind it up really fine. You want to have good particle statistics for X-ray diffraction, and then dropping it onto a silica zero background sample holder to try and get that flat homogeneous surface that's so important. And then for those of you who haven't seen XRD data before, this is sort of what it looks like. You've got your 2 theta on the x-axis, intensity on the y-axis, and your observed pattern is in blue. This is using a software called GSAS2, which is an open source code for XRD analysis. And then you're trying to match database patterns of different minerals to this observed pattern. And so the calculated one pattern, that's a conglomeration of all the different mineral phases we've identified, is showed in green, overlaid on top of that observed pattern. So this is an example of one evaporite and one sediment from these lakes. Going through on this evaporite here, we've identified multiple different sodium carbonate phases. So there's a lot of thermonatrite, which is sodium carbonate with one water molecule at the 60% weight percent level. And then for reference, in this sample here, the thermonatrite is this blue. And so each mineral you expect to see different peaks in the 2 theta range. And so we're just matching those peaks. We're also seeing trona, which is another sodium carbonate, as well as thenardite, sodium sulfate, halite, sodium chloride, berkite, which is a sodium carbonate sulfate, and nacolite, a sodium bicarbonate. So these mineral phases are all very common in these evaporites we're finding across these lakes. While in the sediment, we're seeing a lot of feldspars, like albite, augite, as well as anorthoclase and anorthite in other samples, around the 70% to 20 to 30% range. A secondary phase we're seeing a lot of in the sediments is dolomite, which is that calcium magnesium carbonate. And that's very important. I'll come back to that in a moment. We're also seeing a lot of minor phases like quartz and Vesuvianite and other things like that that are just present in the natural soil in the area. So then you do this a bunch more times because you're a graduate student and you do a lot of labor and you take a lot of samples. And this comes together into the complete XRD results from this study. So I've divided this into results for Last Chance Lake as well as Good Enough Lake, and then in each respective lake, the sediments and the salts being the evaporites around the lake shore as well as the precipitates in the lake. On the left-hand side, we have all the different mineral phases we've identified. So in the sediments, the main phases of interest were these feldspars, both plagioclase feldspar as well as alkali or K feldspars. As well as dolomite, those calcium carbonates, and mica, the clay minerals. We're also seeing some thermonatrite, that sodium carbonate, in the Good Enough Lake sediments, but not in the Last Chance Lake sediments. And then the main mineral phases we're seeing in the evaporites, or the salts, are those sodium carbonates, both thermonitrite and trona, as well as berkite, that sodium carbonate sulfate. A quick word about hydrated minerals. We only detected thermonatrite, which is sodium carbonate with one water molecule, in the XRD patterns after sample preparation. However, previous studies of this area have identified natron salts as the predominant phase. Natron is just sodium carbonate with 10 water molecules. And so if the relative humidity is below 60% at 0 degrees Celsius or 70% at room temperature, then natron will lose its water and turn into thermonatrite. And so we're just anticipating that's what happened in our samples here, since the relative humidity in indoor environments is around 50%. So you can see this plot on the right-hand side from Haynes et al. Showing natron here turning into thermonatrite when you've decreased relative humidity and increased temperature. So in summary, we found that the evaporites, or the salts, are dominated by sodium carbonates, sodium carbonate sulfate, and sodium chloride, demonstrating the predominance of that sodium ion in the lakes, whereas the sediments are dominated by carbonates, both dolomite and anchorite, as well as feldspars, plagioclase and K feldspar, quartz, pyroxene, and clay minerals. However, the big takeaway was we found no calcium phosphate, which is that apatite I talked about earlier, in any of the samples. And this was confirmed by ICP-MS data showing very low abundance of phosphorus in all of the different samples we've taken. So it's not just an XRD fluke, it's also backed up by elemental abundances. These results are consistent with the hypothesis that calcium carbonates precipitate early in the mineralization sequence of these lakes, thus decreasing the dissolved calcium concentrations and allowing phosphate to accumulate to high concentrations instead of precipitating out as apatite, which is that calcium phosphate. So phosphate's accumulating to high concentrations in solution, and that's great for that prebiotic phosphorylation that you need very high phosphate concentrations to get that incorporation into your nucleotides and the potential origin of life. So these could be really good environments for analogs for Hadean Earth lakes where life started. And that's it.
Thanks, Kimberly. We have plenty of time for questions. I have a quick one for you.
Yeah.
Do you know how Good Enough Lake was named?
I think it was something about westward expansion, and when they got there, they were like, oh, this is good enough, and then they turned around and went back east.
Yeah, I was just curious, because when we were in Australia, there was a lot of strange lake names out there, like Dead Kangaroo, Pete's Piggery, and we didn't really understand like the history of them, but
Yeah.
anyway.
There were a lot of jokes in the field about, oh, it's our last chance to get our last chance sample and stuff like that.
Hi, I'm Ellie from CU Boulder, and I was wondering if so I know you said your sediments are plage, dolomite, mica, and feldspar dominated, and I was curious if you would think that the similar pattern that you saw here would happen even if your soda lake was hosted by a different type of like base rock. essentially? Like if we change, is it basaltic or things like that? Like, do you feel like the same pattern would hold in terms of the precipitation of the evaporites?
Yeah, these lakes are very dependent on the host mineralogy, and so you wouldn't expect to see the same aqueous chemistry in a different host environment. And that's one of the reasons why this environment is so interesting, is because of that plagioclase base rock causing this very specific aqueous chemistry. So even just like, 100 miles nearby is an incredibly sulfate-rich lake that people study for different reasons. So slight change in geology, big change in aqueous chemistry.
Hi, really interesting talk. I have kind of two questions. You mentioned that you collected samples seasonally and did sediment by depth measurements. Do you see any variability in your mineralization based on like seasons, and was that reflected in any depth transect?
Yes, we hoped we would see more variation, but we didn't. The only variation you really saw was that you could say, and maybe this isn't even a statistically like significant conclusion, is that you might see more dolomite at the top of the sediment core, so like right in the interaction zone with the water. And you're getting less dolomite at depth, which makes sense for calcium carbonate precipitating out of solution, whereas at depth in the sediment cores, you're getting more of those feldspars, which is just the host rock. But not as much in terms of seasonal variation.
Hi, I'm Shiva Agrawal from Western Michigan University. Can you go 2 slides back? Yeah, so like you have data for, I think, nacolite and thernandite, which has uncertainty higher than the data. So is it normal, or how can you explain this?
Sorry, could you say the last part again?
So the uncertainty in the data is higher than the value? So can you explain this?
Yes, because a lot of these samples only have these minor phases in a couple of the samples. And so there's just not very good counting statistics. We're talking about maybe 3 out of 100 samples or something that have these minor phases. So, yeah.
Taylor Plattner at Georgia Institute of Technology. This is really cool. I had no idea that these lakes were so high in phosphate. I know a couple of people that have studied like Last Chance Lake and then the Basque Lakes. I was curious because you were saying at the end, interesting in terms of like the origin of life. Have you thought about, actually I have a couple questions. Have you thought about like looking at what may be preserved in these salts, and I also have a question on, I know you did XRD, do you plan on doing any other analyses with your samples?
Yeah, so I guess your first question about if we're studying the present life in the lakes? Yeah? Yeah, so at a macroscopic scale, when you go there, it's really interesting because you see all these brine flies that are just encrusted in the salt. Which is kind of freaky, because in the summer you're like encompassed by flies, and then in the winter it's like a fly cemetery and there's just a bunch of dead flies on the ground. But in a more astrobiology sense, there are other teams at, I think it's, University in Canada, who are studying the current microbiology in the ice, because there are living microbes in the ice in the winter. And so we're not necessarily doing that work, but there are colleagues who are. Wait, what was your second question? Yes, okay. So my section of the work was a lot about the mineralogy of the sediments and the evaporites, but my colleague Sebastian Haas did a lot more of the water chemistry. And so we're submitting a paper now, and hopefully you'll be able to read that and hear all about his work as well.
Thank you very much, Kimberly.
Yeah.
Okay, our final speaker of salty stuff, if PowerPoint will work, is Chad Pozoriski from Georgia Tech.
All right. So I'm going to call back a little bit to a couple of the sites that Emily mentioned before. And what's exciting for me is that I'm in San Diego for the first time, so I finally might be able to go see one of the field sites I'm about to talk about. And I had no idea that there were actually palm trees down here around my supposedly extreme field site. So that'll be a funny dichotomy there. But so yeah, we're going to be following the salt, and it's going to be a lot of salt. So hang tight there. So the first field site, South Bay Salt Works, which is down the road, is a salt farm, which as water is taken from the bay, it is brought in, and the people there are basically taking the halite out and using that to, I believe, soften the water here. And then the magnesium chloride also gets concentrated and separated. And so you get these really sodium chloride concentrated regions and magnesium chloride concentrated regions. And then you also have regions which are a lot more like seawater. And so that's South Bay Salt Works. That's one of the sites I'll be talking about a lot today. And what we're investigating there with oceans across space and time is, can biosignature molecules be detected in these sites with these super high salinities? And then how does that impact our search for life elsewhere? And then how do these vary with things like water activity and ion concentration? And then here's another analog site in Western Australia. This is Lake Campion, which is a very interesting site that we looked at. So these are transient lakes, and so they evaporate during the drier, warmer months, and then they fill up during the winter months, and so you get these salt crusts which build up on the bottom of them. And so these are really interesting because the evaporation process is happening very actively, so we can get a modern look at what's happening there. And so in general, analog sites can give us training grounds for robotic exploration on other planets. They can help us test our instruments, and particularly we want to know how salt and acidic low pH environments affect our instruments. And then it can give us insight into the environmental processes and the biology happening there. And so now I'm going to shout out Luke Fisher's review paper in Environmental Microbiology, where he talks all about biosignature preservation in brines, particularly in deep hypersaline anoxic basins, which I won't be talking about today. But when it comes to brines themselves, as opposed to evaporites or salt crystals, we know that DNA and rRNA are well preserved, while mRNA is not as well preserved, and that ATP preservation has been observed by Tuovola et al. In 1987, but not much has been done about that since then. And lipid preservation is being studied more and more recently, and so that's good. But these aren't nearly as well explored as genetic compounds. And so one compound that we're looking at, ATP, adenosine triphosphate, it's a short-lived molecule, and it's used in astrobiology and in ecology as a marker of microbial activity. And so if you have a lot of ATP, it means that a lot of microbial activity is happening. If you don't have any microbial activity, then you're probably not going to have much ATP. But it's very highly evolved. So as a biosignature, it might be less agnostic than, for example, amino acids. And so we could look at polypeptides, or we could look at individual amino acids, and we target biological amino acids in this study, as well as osmolate amino acids, which are solute compounds that are accumulated by microbes when they're under high stress in hypersaline sites. Yeah. So, and that's well understood in the literature that these osmolates exist. And so first I'll talk about the methods that we used in the field, or near the field in the motel room. We looked for ATP using a luciferase assay. And so say you have a beautiful crystal from the crust of a lake like this from Western Australia, then you take it to your Western Australia motel room and you do you set out your samples, we weigh them out, we extract them in hot water, and then we add our luciferase and luciferin, and we have a field portable luminometer in order to do our analyses. And so that's how we quantify ATP. And moving on to what we do at Georgia Tech when we bring the samples home, So I was so glad to hear that another person here was using capillary electrophoresis, so I don't need to explain it in its entirety again. But it's a separation method that in our lab we pair with laser-induced fluorescence. And so laser-induced fluorescence means that we're tagging our amino acids, our compounds of interest, with a fluorescent dye, so we're targeting specific compounds. And so this is a targeted technique which has very low limits of detection and very high sensitivity. And so we did this at South Bay Saltworks on one of the samples that has a water activity of below 0.4 and magnesium concentration of more than 4 molar. And this is only a 1 to 10 dilution. So using a method that Marshall Seaton and I developed, we got the concentration of amino acids in this sample down to tens of nanomolar. The limits of detection for this method in general are in the tens of picomolar. And we did this using a commercial instrument. And again, it was only a 1 to 10 dilution for this hypersaline sample. So we were delighted that the results came out like this. And then for an untargeted analysis, we can use microchip capillary electrophoresis paired with high-resolution mass spectrometry. So if we take the same sample and we look at it using an untargeted method where we can get the mass-to-charge ratio of the compounds that we're interested in, then combining the zip chip commercial setup with a thermo-Q exactive Orbitrap, this is what the chip looks like, it's very cute. We get a very interesting set of compounds which we didn't necessarily target. And so moving back to the LIF analyses for the results, what we first observed was that as we increase in magnesium concentration and decrease in water activity and increase in chaotropicity, we get a significant increase in the dissolved free primary amines that we were targeting. And so that would be our biological amino acids, which you've identified here. And as you go up, you have higher magnesium sites, and we have a lot higher peaks, and we also have a lot higher values for quantified amino acids. And so we attribute this to evapoconcentration, as the sites which have a lot of water and a lot of salt and then evaporate and leave the salts and the organics behind, the concentration is higher. So that's pretty straightforward and simple, but it is very useful in the case where we're looking at very low biomass regions. So next, we then put it in microchip CEMS, and we looked for these same compounds, and we found a lot of them there. And so you can see histidine, glycine, alanine, isoleucine, and leucine are resolved there, serine, et cetera, glutamic and aspartic acid. And this was only a 1 to 4 dilution of that same sample. So we were really delighted by these results as well. And in the same separation, just looking at different traces, different M over Zs, we see a couple polypeptides, and we see some adenine and guanidine in there as well. And so these are things that we didn't necessarily look for in the CELIF, but now that we're combining it with an untargeted method, we can get a look at some more compounds that we didn't necessarily expect. And then also osmolytes, which we did expect and did want to look for. We found those like ornithine, sarcosine, and betaine in our samples with very high salt stress being applied to any of the microbes that were alive there. And so these are there in significant quantity. And in Western Australia, briefly, I'll just show you some preliminary results. This is Lake Brown, which has a sodium concentration of near 5 molar and a water activity of 0.89, so not nearly as low a water activity as the South Bay Saltworks sample I just showed you. But it has high relative concentrations of a lot of these compounds, including the osmolyte compound betaine, and some lower concentrations of other compounds, including a polypeptide. And then conversely, at Lake Gunter, which has a higher water activity and a lower sodium concentration, we see that there really isn't any easy way to detect those organic compounds with the method that we're using right now, so just diluting these samples. So perhaps this indicates that and of course this is only 2 data points with Western Australia, but looking at what we found at South Bay Saltworks, perhaps these saltier sites are actually places where it might be easier to detect biomolecules because of evapoconcentration. And we also want to look at the normalized abundance of certain amino acids just to see what sort of patterns we could find. And if we have water activity increasing from left to right here with the magnesium sites here, we see that certain amino acids are upregulated in this magnesium heavy site and others are downregulated. And with that information, we decided maybe we could classify our sites based on the amino acid distribution. So if you take the distribution in, you put it in a classifier, either logistic regression or random forest, you could get out one of these 3 different site types. And this is a small data set and more of a proof of concept than anything, but it's interesting to see that we have a very good area under the curve of 1 or 0.83, which, and we also can see which compounds are driving that the most. And so that's an interesting thing to take note of that might be used in the future if we have much larger data sets. And so the last thing I wanted to talk about, ATP, if you remember. So on the x-axis here is magnesium concentration, and then we have 3 y-axes, because why not? And first of all, I want to mention the cell counts by microscopy, which is from Ben Klempe, showed that the number of active cells in these sites go down as you get to the super high magnesium concentration regions, which is also what Emily showed in her talk. And so what we then can look at is the ATP concentration, which strangely goes up, even as the concentration of active cells goes down and the expected activity goes down. And so we wondered, why is that happening? Because as we know, the amino acids are concentrating, but amino acids don't decay on really short timescales. They're not broken up by hydrolysis or by ATPase enzymes. But what we think is that something must be driving this, and if it's not evapoconcentration, we're wondering, since the ATP hydrolysis rate is much quicker than the evapoconcentration rate, why would the ATP accumulate? Doesn't really make much sense, but perhaps it's being preserved. So this Tuovola et al. Paper from 1987 that I mentioned way earlier, they found that ATP was preserved in low water activity sites. And so that could be preventing the enzymatic and natural hydrolysis of ATP, leaving it to persist in the solution. And additionally, it could also be used as an osmolyte compound as well and be accumulated by those microorganisms before they end up in a super high magnesium site and not very active themselves. So, in conclusion, we can detect biosignatures at micromolar concentrations in near saturation brines using CELAF and micro-CEMS. And amino acid distribution ratios can be well classified based on their brine type. And osmolytes are present and detectable in some but not all of the South Bay saltworks and Western Australia brines. And we found preservation of ATP at South Bay saltworks. And so basically my argument is then, why don't we follow the salt and see if we can find biosignatures there on other worlds. So thanks for listening.
Thank you, Chad. We have time for 2 questions.
Chad, thank you for the talk. That was a beautiful separation that you showed earlier. So I'm curious. You did a quantitation of amino acids using CELAF method, and it looks like you've done some work using CEMS. Have you tried quantifying the amino acid content in the same sites using microchip CEMS and seeing what those look like to try and cross-validate both methods and using the same sample yet, or have you not got to that yet?
Nope. So we just started doing the micro CEMS work over the past month, and so we're really happy how it's been doing with qualitative untargeted work, but we haven't gone into quantitative because the ion suppression effects might be a bit of a challenge to overcome, so that'll probably be a little bit of a longer project. What we have done to try to corroborate the amino acid concentrations is look at the expression of, or not the expression rather, but the presence of certain proteins in the genetic code of the microbes that were found to be present here by other OST collaborators. And we find a moderate correlation between the amino acids we see and the amino acids that they see, but since we don't have transcriptomes from all of these sites, we don't know exactly what proteins are being made. And so I think that's the next step is to connect those from metabolomics to transcriptomics to genomics, just as the talk last night was discussing. So, yeah.
Thank you.
Any last question for Chad?
I have two questions actually.
Yeah.
Maybe I didn't really catch you. What's the pH of the side that you show that magnesium concentration is more than 4 molar?
That's a great question. I don't know off the top of my head, but I can get back to you in a moment. Yeah.
Okay. And also, how would you determine your ATP is accumulated or protected under high saline water or like low water activity?
How did I, sorry, say again? How did I?
How do you know that your ATP is not hydrolyzed?
So, yeah, I don't know that it's not hydrolyzed necessarily, but I am predicting that. So the typical destruction method of ATP, either in the cell or in the environment, is through hydrolysis, either performed by proteins or just happening naturally in the environment with water. And so those are the things that must, from what I'm observing, have been stopped if the ATP is going to persist so long for it to be evapoconcentrated to those high of levels. The huge spike there likely wouldn't have been possible if evapoconcentration weren't a component. And so, unless the osmolyte effect is so much more significant than I had thought, that could be another reason. But I still think that the preservation of ATP is linked to the lack of a rapid breakdown mechanism.
Have you ever considered that ATP with divalent cations form some nanostructures to make the things happen?
Yeah. So whether it stabilizes it in some way, So I don't honestly know much about how I would investigate that. I've spoken to someone recently about potentially modeling it using molecular dynamics, but I haven't, waded into the actual the structural chemistry of whether that would happen. But if you have any ideas, I'd love to hear them. So, thanks. Yeah.
Thanks, Jed. Okay, so that concludes all the talks for today. We have a extended poster session, so it's a little bit longer than the others because we have some excellent local undergrads that you'll find outside on the back patio. And thanks to a group at Upper Campus, the Cosmic Tours, we have an indoor planetarium that's a few doors down. Miguel just posted a little map of where it is. So we encourage you, throughout the poster session, see if you can get in there. I think it's about a 20-minute tour or so. And besides that, we're all going to meet at Shore Rider at 7 o'clock. That's when we'll start serving dinner. And then trivia will start right at 8. So just feel free to relax at the hotel or something before. And let Miguel or I know if you have any questions or concerns. Thanks.